Hi again. Most of you guys won't have any idea because you just see this recording, but if you watch the intro to this lecture, I was in a different place, and if you look outside here, notice it's dark outside now. I was using Panopto for the first one, gave me a little grief, no problem. Did the whole lecture and went to look at it, and Panopto had stopped recording and didn't even have the courtesy of telling me that it had stopped recording. I'm espousing these days the beauty of stoicism and yoga and being peaceful, inner peace, and all this. This is fate testing me if I can actually walk the walk rather than just talk the talk. So anyway, I'm back. You guys wouldn't notice all the behind the scenes, but I am not a fan of Panopto as it is right now. So I'm just checking, at least now I'm using Zoom. I've switched formats. Zoom, I can see if I'm recording here, it looks like it's recording. So this is my second trial. I just chalked the first one up to a practice run. It was probably way better than anything that you'll see in this one. So I'll just get into the lecture. So hopefully this is screen sharing. Hopefully you can see this. I have two options now. I can either keep going like this or actually stop every 10 minutes and check if it's recording. I'm feeling reckless, so I'm just going to go with recording. We'll see what happens. So if you didn't get a chance, I'd encourage you to maybe briefly look through the intro to the whole course that I just did a few hours ago. Right now, I'm going to give a lecture on drug targets. So this is an introduction to chemical pharmacology, this supplementary course in chemistry. My name's Grant Churchill. I'm in the Department of Pharmacology. I'm also at New College, Oxford. I'm the organizing tutor for medicine and biomedical sciences there. If you don't listen to the other one, I'll just briefly say my research interests and my interests, what I read in my free time, my spare time, have to do with chemicals and biology and how chemistry can explain biology. And I'm very passionate about this. I started this course about 2014, 2015, something like that. And this year it's a little different. If you go to the last lecture, you'll see, last outline of the course, you'll see that I'm just providing lectures this year. I'm not involved in the organization, happily. So this is going to be about drug targets. So drugs are small molecules, usually, historically, that's certainly changing nowadays. They bind to so-called targets. So what's that all about? So I pose this first question. This is a blog by this gentleman up here, Derek Lowe. And his blog's called In the Pipeline. Very often drug discovery is described as a pipeline process. And Derek writes this blog. He works, I think he worked for Vertex. I'm not even sure which pharmaceutical company he's at now. He's in the Boston area. It's just an area that's bubbling with new biotechs all the time. A lot of the very important biotechs now started in the Boston area. So it's a great atmosphere. We're striving to recapitulate some of those things at Oxford, Cambridge, London, Golden Triangle, and all that. We haven't got it down just right yet, but it's a great area. So he's got his finger on the pulse of a lot of this. If you're interested in pharmacology, drug discovery, I'd encourage you to look at his blog. He blogs every day right now. It's mostly coronavirus stuff, rightly so. I guess none of us can completely get away from that. This blog was in, let's see, up here, if you can see my cursor, in 2018, March 2018. And I was talking about a number of drug targets for a class of drugs called Nsaids. So I've circled those down here. Nsaids are non-steroidal anti-inflammatory drugs, or Nsaids. This includes drugs that you'll probably be very familiar with, such as aspirin, ibuprofen. These are over-the-counter drugs. You can go down to Boots and buy a bunch of them. And people have known how they work, or at least we think we know how they work, since the late 60s, early 70s. There was a guy who was associated with the Department of Pharmacology called John Vane, Pharmacology in Oxford, the department that I'm at. And he discovered that aspirin, its main effect seemed to be through an enzyme called cyclooxygenase, and I'll describe that later in this lecture. Right now, I just want to give you an overview of some of these ideas. And this was such an important discovery that he got a Nobel Prize for it. So you might be surprised that somebody could get a Nobel Prize for that type of discovery. Nobel Prizes were awarded for the first demonstration of a receptor. People used to think that receptors were just figments of pharmacologists' imaginations. They were useful constructs, but they had no bearing in physical reality. We now know that's not the case. What about the idea that receptors, or in this case, one of the receptors or drug targets, I guess more technically, is a cyclooxygenase for oxygen? This is what all the textbooks say. This is what everybody says. This is what I lecture to the medical students on and the biomedical sciences students on. In my heart of hearts, I used to probably even believe in it. It's definitely the target for certain things. So cyclooxygenases are involved in producing something called prostaglandins. That's down here. Prostaglandins give you fever. They give you inflammation and redness, and they give you pain as well. So fever, pain, inflammation. There's some Latin that I forgot. If you're a medical student, you'll remember this. If you know medical students, you can ask them about it. It's like rubor something or else, or all these things. It's a cute little memory device that I can't remember, which is kind of ironic. Anyway, so we've got cyclooxygenase makes prostaglandins, pain and inflammation. Aspirin inhibits this. Guy got a Nobel Prize for it. Seems to be a done deal. But is that the case? I'll tell you more about cyclooxygenase 2 inhibitors later. A lot of enzymes and receptors come in families. They have different isoforms, different flavors, if you will. We can then drug different ones because they have slightly different preferences, slightly different binding sites. So I'll talk to you more about that later in this lecture and in the next lecture as well. What this group did, and what Derek here blogged about, was they took aspirin and they made a covalent group on it that would bind to the targets. I can't remember which one they used. You can put like an azide on it, N3, 3 nitrogens together. You flash it with UV light, it takes off, and you covalently link your aspirin to whatever. Then you can do proteomics, any omics. It's all the rage these days. You run it over a mass spec, it gives you information, and you can try to figure out what aspirin's binding to. They didn't get just aspirin binding to cyclooxygenase 1, cyclooxygenase 2. They got in one cell line, this K562 cell line, 513 targets. They ran another cell line, a JURCAT cell line, that's down here. And I can't remember how many they got, but there were 206 that overlapped. But I think they got about 500 in the JURCAT cell line as well. So this old drug, acetylsalicylic acid, aspirin, that we all think inhibits cyclooxygenase, and that's the way it works. It might work like that in some instances. It seems to bind to a hell of a lot more protein targets. Are those protein targets just chance and don't do anything? Are they neutral? Are they silent? Or are some of these targets important? There's other data that I don't have time to get into that shows some of these other targets might be important in some of the aspects of aspirin as a pharmaceutical. So this is an interesting idea because everybody in drug discovery, everybody that teaches pharmacology, all pharmacology textbooks talk about drugs and their target. This whole lecture is going to be about drug targets. So the underlying foundation premise is that one drug binds to one target. Might not be the case. In fact, especially with something like central nervous system drugs, drugs of the brain, These probably have multiple mechanisms of action. We call them polypharmacologic mechanisms of action. And it's the way things really work, but people seem to like this fiction that you have a single drug, a single target. A disease has a single defect. We can drug a single target, cure the disease. It's probably an oversimplification. You got to start somewhere, but it's something to think about. I'll tell you more about this story as I go along. Each lecture, I'll give you a little snapshot like this. It's like, think of these things as tabloid headlines. This is like the Daily Mail version of drug targets. So, oh, one thing I forgot. Down here, I love this. He writes very poetically, as far as I'm concerned. So if you needed a reminder about just what a complicated mixing bowl we're throwing our compounds into, here you go. So this complicated mixing bowl, that's basically a cell. That's biology. Biology is defined by cells. In terms of cells, something that might surprise some of the chemists out there is that most of biology, all the chemistry occurs, most of the chemistry, 99%. So in biology, there's no absolutes. That also is something chemists should be aware of. There's no absolutes in biology. You try to make generalizations. 99 point something percent of biology occurs, the chemistry associated with biology occurs in the aqueous phase. Most chemistry, at least traditionally, has been in the organic phase. And these two phases don't mix. And you go to conferences and people still make jokes. The old ones are the best ones, sort of thing. Oil and water don't mix. Chemists and biologists don't mix, all of this. This course in chemical pharmacology is going to attempt to get oil and water to mix. The chemists and biologists, it'll give chemists a way to think about biology conceptually. I don't have time to give you all the details, but conceptually you can hopefully cover what's going on. And the biologists, hopefully I'll be able to give some bit of chemistry so that they can understand, they can look at a molecule and understand how it works, even make predictions, and then go to the experimental evidence to see if that is the way it actually is working. So in terms of, I'm just going to actually kill the video and make sure it's saving. I'm getting all paranoid now. Check the video. It looked like it was recording, so we are good. I'm good. You guys probably don't care or know. So everything's working. So I was using Zoom though, I noticed that moving my little thing around here, the picture of me, it will cover up part of the screen, unlike on Panopto. This could be a problem, but hopefully you'll have the PDF up there. So if it's covering something while I'm going through this, you can look at it. If it's really obstructing the view, hopefully I remember to actually move it. Where I was, to resume, is that this is a cell. Everything's aqueous except the interior of the membrane. Most of biology occurs in the aqueous phase. Very little occurs in the organic phase, the hydrophobic phase. This membrane delineates the cell. This is a cartoon of a cell. There is an outer membrane called the plasma membrane. Then we have membrane-bound so-called organelles, mini organs, organelles, inside the cell. This compartmentalization is for function. The mitochondria is one organelle. It's sort of stereotypically the powerhouse of the cell. It makes a highly energetic molecule, ATP, which is the energy currency of the cell. So that goes around there. The nucleus has a membrane around it, actually a double membrane, because that was originally a bacteria that got engulfed by another cell. And so it has a double membrane. There are multiple organelles inside here. The ones I want you to think about, you don't have to just laundry list memorize these, but know that they're all subdivided by membrane, the cell's subdivided by membranes into these organelles. So you have a molecule and you want to get it across. If it's a drug, you're trying to get it to its target, which this lecture is about. If it's a metabolite, you're trying to constrain that metabolite. So the idea is that the origins of life, to get philosophical and sort of evolutionary for a second, origins of life, in the primordial sea, there were waves lapping up against the shore, making a bit of foam through some phospholipid molecules. I'll show you what those are in a minute, basically making bubbles. Bubbles are inside out cell membranes. I'll show you why that's true in a second. The reason that we think that was necessary for life is you have to compartmentalize reactions you want from the outside world, from the environment. There's you and there's the environment, and they're different things. You want to keep molecules in there, probably polymerize them, probably copy them so they're the same polymer. This is what we think the origin of life was about. It has to do with making these bubble-like structures, which are membranes. So everything in a cell is aqueous except the interior of these membranes. So on this next slide here, what I'm showing you, ideally I would give you like 4 lectures, 4 one-hour lectures just on biological membranes. They're fascinating biological structures at every level. I'm limited to just a couple slides. So this lecture, what I'm trying to do is give an overview of biology to chemists so that they know where drug targets are, what these biological macromolecules are that we call drug targets. I'm going to put them in the context. I'm also going to talk about membranes because these molecules have to hit their target. Their target could be in the membrane or it might be through a membrane. So membranes are important. And then also just fundamentally, they're important for the organization of biology and the origins of life. This is an electron micrograph of a membrane. This is an artist's cartoon conception of a membrane. We now call this conception the fluid mosaic model. It's called fluid mosaic because there's 2 planes. It's a bilayer of phospholipids. They're depicted diagrammatically or cartoon-like down here. And these 2 bilayers can slide. Each phospholipid within the bilayer can move, but nothing can flip across. So it's a fluid. It's fluid in this plane, in 2 dimensions, but it's not movable in 3 dimensions. Things don't flip. There are some enzymes, but we won't get into that. Things don't really flip across the membrane. There are all sorts of experiments and ideas. Biology is a science where you have to do hypothesis testing. This is not just philosophy. The ancient Greeks did some, what they thought was science with Aristotle and that. And it was just like, why bother doing an experiment? You just think about it. Nowadays, there's many hypotheses that could fit how things work, but we have to test them. As this guy, Richard Feynman, the Nobel Prize winner for quantum electrodynamics, is famous for saying, amongst other things, is that you have a hypothesis, you collect data. If the data don't fit your hypothesis is wrong. Doesn't matter who you are, doesn't matter how many degrees you have, how many letters, doesn't matter how elegant the idea is. If your hypothesis doesn't fit the data, it's wrong. So there are a lot of hypotheses surrounding membranes. A lot of them are partially right. This is the way science works. You have a hypothesis, you get data, you say which bits fit your hypothesis, which don't. You regenerate a hypothesis, you move on. It's fun. It's like a detective game. That's why some people like me are into this. So the definitive evidence, I won't take you along that whole pathway, as fascinating as it is, I'm going to give you some definitive evidence. This is an electron micrograph. So you shoot electrons at tissue, a thin slice of tissue. This is a transmission electron micrograph. And if things are electron dense, the electrons scatter, and they'll be darker with the way this works. They'll be darker here. So the dark bit is inside a cell. Outside the cell, it's clear. This is not just macromolecules. It's treated with very often salts of uranium, so uranyl acetate, these types of salts, and they will stick to macromolecules. Uranium's very heavy element. It's got a lot of electrons around it. It'll scatter electrons really readily. So that's what you're looking at here. The thing I'd like you to look at is this very edge of the cell. If we zoom in even more, we'd see train tracks. Hopefully, if you squint, you can even see sort of this railway track structure along here. And that's because these phospholipid head groups are electron dense. The hydrocarbon tails, these are fatty acid acyl chains in the middle, the hydrophobic, the oily bit that I told you about that bound cells and the cell organelles. It's less electron dense. So you actually get this railway track appearance. There's a few other EM things. Go into a textbook. For a lot of the biology that I'll be talking to you about, go into Albert's Molecular Biology of the Cell. It's probably the most accessible and clear from a chemical perspective biology, especially the first few chapters. They've got some beautiful diagrams in there where they go from molecules to structures and what the molecules mean. Then we can take all that basic biology knowledge, biochemical knowledge, and overlay pharmacology onto it. It's a very good starting point. And they do a good description of membranes in general with all the history. So this is good evidence. We've got this lipid bilayer, fluid dynamic structure. The fluid is very important. If the membrane's not fluid, these proteins inside here, so here's what we call an integral membrane protein. It's also diagrammed down here at a larger scale. This integral membrane protein has a hydrophobic bit through the center and hydrophilic bits on either end. These can be channels that allow small inorganic ions across the membrane. They open up and form a hole so these ions can flow through. Or they can be transporters where they take metabolites, small molecules that wouldn't pass through a membrane normally, grab them, transfer them across the cell. These can be passive or they can be active, meaning they can actually concentrate some substance against a gradient using energy. If this membrane, if these hydrophobic tails are not fluid, the proteins stop working properly. I'll give you some examples in the next slide of that. So the overall message from this slide is biological membranes are what delineate cells and the cell subcompartments. Metabolites and drugs are very similar in this respect. Some pass passively, some of them have to be transported to get across. I'll tell you more about the molecular properties in a minute. And this biological membrane is not covalently bound. This is pure thermodynamic forces, mostly hydrophobic interactions, causing all these tails to go together and all the polar head groups to stick out to the side. So that's what's driving this. It's a key concept in biology. In terms of membrane lipids, so if we just go back for a second, this head group and the tails, the head's hydrophilic, the tail's hydrophobic, polar, non-polar. If we pick now on one of these, so there's a lot of different phospholipids. I'm going to tell you about one phospholipid and then I'm going to tell you about cholesterol. But that doesn't mean that there's not other ones. Phospholipids, their chemical structure, their backbone, even though it makes up a small part of the overall molecule, their backbone is made up from glycerol. Here's glycerol. 3 carbons, 3 hydroxyls, 1 hydroxyl on each carbon. Then they are, excuse me, the glycerol is then, each hydroxyl is esterified. 2 of them are esterified to fatty acids. So these form fatty acyl groups that are those chains that make up the hydrophobic bed of the membrane. The third hydroxyl on the glycerol is also esterified. Rather than to a carboxylic acid, though, it's esterified to phosphoric acid. Here's phosphoric acid up here. There's a phosphate. So that's why these are called phospholipids. Then there's a head group, and the head group can be one of several things. In this case, the head group happens to be choline. So 2 carbons, a quaternary ammonium nitrogen, and choline itself would be an alcohol. So an alcohol with an acid gives you an ester. That gives us this bond up here. So structurally, this is the big circle that we draw, and these are the 2 legs in that previous slide's cartoon. That gives us that structure. And the chains have to be flexible enough so that the integral membrane proteins in that membrane can actually move to do their job. There's conformational changes in these proteins so that they can transport things, or open if they're ion channels, or do a conformational change if they're a receptor to hormones and neurotransmitters to convert the signal across the membrane. Even though I've just given you one example here, this happens to be called phosphatidylcholine. This head group can vary. You could have ethanolamine, you could have serine, you could have various carbohydrates like glucose. All of this is there. You can have inositol, a 6-member ring with hydroxyls at each point. Then it can be decorated with phosphates at each hydroxyl position. All of these are phospholipids. All of them play structural roles in membranes. All of them also, and this is a key point, as I say at the bottom of the slide, not just structural, but also signaling roles. If we take this phospholipid and we hydrolyze it at any one of these esters, we will get products. Those products are signaling molecules. This is what we call a process we call signal transduction. So there's very specific positions. Any one of these that you hydrolyze, you'll get signaling. The one we're going to pick on today is this position. It's called, this position, the A2 position, so-called A2 position. The fatty acid here is a specific one. It's arachidonic acid, and it will get metabolized into prostaglandins, which I told you about in the first slide. That enzyme that then converts arachidonic acid into prostaglandins is cyclooxygenase. So that's the link here. But you can get any cuts here, any hydrolysis, you will get molecules that signal. Very, very powerful. And they go through amplification processes so that just a few of these molecules will give you a very big response later down the line. I'm just going to give you in this lecture very specific examples, and it's up to you to extend them to other things. You won't have to know it for the course, for the exam and things, but in your life, if you're a medic or a chemist or a scientist eventually, or just a person who goes off and does banking or something and is reading about coronavirus vaccines, messenger RNA versus others, these types of things give you the ability, the skills to think about chemistry and how it impacts on biology from first principles. That's what I'm trying to do here. So I'll give you examples and then you can extend them to other things. So phospholipids, many different types. They play structural roles in this membrane. Hydrophobic forces them into a bilayer, but they also play signaling roles. Any of these ester groups that get hydrolyzed will cause signaling molecules. The other molecule I'd like to mention now, because it will be important in terms of signaling as well as a structural role, is cholesterol. So normally cholesterol is drawn like 90 degrees anti-clockwise to what I've done here. I've spun it 90 degrees clockwise so that you get the idea that it has the same hydrophobic, hydrophilic orientation as a phospholipid. Cholesterol is very greasy. It's all hydrocarbons other than this one hydroxyl. So in a lipid membrane, it inserts that way. Different membranes throughout the cell have different amounts of cholesterol. The plasma membrane, the surface membrane, has around 1%, 1 to 5%. Other lipids are very deficient in it. They'll have very, very low percentages of cholesterol. Cholesterol affects this fluidity of the membrane. You can see it's a ring structure along here and then a floppy chain. So if you insert cholesterol, you disrupt the packing. Normal phospholipids have these fatty acid groups, these fatty acyl chains. And if they're all packed together, you will decrease fluidity. If you put kinks in them through double bonds, you'll increase fluidity. Double bonded fatty acids can't pack. If we have one with a double bond, it's kinked, and one isn't, it will distort the packing pattern when you're trying to make crystals of this. So membrane fluidity can be controlled with fatty acid oxidation, double bonds, or it can be controlled by inserting cholesterol. Cholesterol is a little trickier. It varies with certain things, but for our purposes, let's just say cholesterol makes the membrane stiffer. Within reason, that's not such a bad thing. So now let's look at a slightly different aspect of this. We'll look at fatty acid in a little more detail. I said there's 3 hydroxyl positions that can be esterified. One of the positions, the third position's always got phosphoric acid on it. The second position very often has this fatty acid, and it's kinked. And this is called arachidonic acid. It has 20 carbons, it has 4 cis double bonds. Most of biology is always cis double bonds, and it gives us this kinked shape. So it disrupts the membrane. It will cause an increase in fluidity. In contrast, the fatty acid out here on the so-called A1 position is 18 carbons, meaning it's stearic acid or stearate. Stearic acid is what you would find in butter. Some of this stuff is very useful to think about things in different contexts. Again, I defer to Richard Feynman for a lot of this. He's very much influenced my thinking on these things, and I aspire to do that. It's like looking at the moon and wishing you were there. Feynman was an incredible intellect, but I can steal some of his ways of thinking. Feynman wrote that when he was a kid, his dad sat him on his lap and they read the Encyclopedia Britannica. And as an example, he said they'd be talking about dinosaurs. His dad would be reading something about dinosaurs, T-Rex, and they'd be in the second story of their house. First floor if you're in the UK, he was in the States. And he said, okay, let's think about what this means. They gave sizes of the dinosaur, 25 feet high and a head 6 feet. So he said, if the dinosaur was outside the room we're reading in, he would be able to look into our window, but his head wouldn't quite fit through the window. So Feynman said, always put things into terms that you can understand. And he did that with everything. He came up with a very simple explanation for superfluidity. When you cool gases like helium down, they have no friction anymore and they'll keep moving and all these things. Everybody was using very complicated things. Ironically, Feynman diagrams to try to solve the problem of superfluidity. He took it back to very basics and said, what's the simplest thing I can do? Very often the simplest thing that works is the best. Occam's razor. Simplest explanation is usually the correct explanation. So anyway, getting back to this, the C18 stearic acid, you guys should be able to think about some of this stuff like Feynman in your normal life. If we were doing this live and all, I could see everybody out here in a classroom, I would ask a question. Where does stearic acid come from? What do you know in your everyday life where stearic acid exists? The answer is in butter, basically, or animal fat, tallow. So if we have our glycerol here and instead of a phosphate on it, we just put another fatty acid on there, it's a triglyceride. Triglycerides from animal fat, very often beef cattle, they have all 3 stearic acids, or they have 2 stearic acids and one called oleic acid, which has 1 double bond, 1 kink. Stearic acid, tallow, stearic acid, when it's esterified to glycerol to form triglycerides that if you can isolate as tallow from rendering down beef products, it has a melting point of probably, I can't remember, like 63, 64 degrees Celsius. So it's very, very solid. Butter has other fatty acids in it that allow it to be semi-solid at room temperature. So you can smear it on your toast. You put butter in the fridge, can't smear it on your toast. If you leave it out on the counter, you can smear it on your toast. Contrast butter with olive oil. What is the fatty acid composition of olive oil? It's liquid at room temperature. If you put it in kind of a cool corner of your room, though, you'll start to see it partition. You will see crystals, you'll see opacity. There will be some fatty acids crystallizing out of olive oil. So it's a mixture of unsaturated fatty acids. That's why it's liquid at room temperature. The fatty acids in olive oil tend to be 18 carbons as well, but they have 1, 2, or 2 carbon bonds, so double bonds. So it'd be oleic and linoleic usually. Linolenic is 3 unsaturated bonds, and it tends to be in linseed oil, flax, and it's a drying paint, that sort of thing, because it'll oxidize and polymerase. Think about these things. A hokey example that just came to mind would be McDonald's. Back in the day, they used to fry their fries, chips, in tallow. Then people were very exercised over beef fat, saturated fats. So these are saturated fats. So they told them they had to fry them in other things. So they started frying them in unsaturated fats. What happens is you end up getting chemical reactions because the sites of unsaturation are chemically reactive more so than the fatty acids, the saturated fatty acids. So these were breaking apart. They were forming volatiles. They were forming volatile aldehydes. The aldehydes were congealing on surfaces. Everything in McDonald's was tacky, sticky. People's clothes got tacky, sticky. Somebody like me with asthma would go into a McDonald's and there's enough aldehydes floating around in the air, it would bother my lungs. So what was very good and noble that they tried to get saturated fats out of the diet from their deep fryers turned out to be a bad idea, not just because of these aerosols, but there's something called trans fats. I mentioned all these fats on arachidonic acid, all these double bonds are cis. In nature, with very, very, very precious few exceptions, all double bonds are cis. If you deep fry in plant-based oils that are unsaturated, you will get trans fats. So trans fats you've probably heard of. They're every 10 years, nutritionists tell us new chemicals, the devil. Trans fats were the devil, have been for probably 15 years. They're, from a biochemical point of view, they probably are not such a hot idea to take trans fats in. They don't really occur in nature. Not that that necessarily means something's toxic or not, but there's evidence that they gum up some of the enzymes that normally would metabolize fats. So that's why they are a hazard. So getting back to this now, that whole sort of aside was just to give you an idea about how to think about these things. Actually, I'll give you one more little aside before I drop this. Plants can't just come indoors and put clothes on when it gets cold outside, but they still want their membranes to maintain fluidity, even in temperatures like this, when it gets below zero. The way they do this is by altering the structure of the fluidity of their membranes by introducing different fatty acids with different saturation or unsaturation. This is called acclimation. Plants that grow in the UK know how to acclimate. The daylight starts going down, they start replacing saturated fatty acids for unsaturated fatty acids. If we take bananas are a good example. If you've ever put bananas in the refrigerator, refrigerators are about 4 degrees, leave them there for a couple of days, they'll turn brownish black. The yellow will turn brownish black. It's because their membranes gel. This is another way of thinking about this from a chemical point of view. It's a gel sol, so gel or solution. If you get a gel, not even just full on freezing out where you get a solid, but if you get a gel, these transporters in the membrane won't be able to move ions and things. So bananas get what's called chilling injury because they can't acclimate. They're not used to the UK. So that's one way you could think about this again. The reason I'm telling you all this is there's a lot of things in life where you can think about in terms of chemistry that you know. And if you start doing this, it's a fun game, and you can come up with ideas, and then you can look up what the real reason is. And sometimes nobody's even thought about this. And that's where it gets really delightful. You will have thought about something that nobody else on the face of the earth has ever thought about. And it seems obvious and simple when you get there, but nobody else has thought about it. Then you do the experiment, and usually you find out you're wrong. But hey ho, that's the way science goes. And it keeps you going for another day. You can generate more and more hypotheses. Hypotheses are cheap. Demonstrated hypotheses are difficult. Anyway, little science philosophy. So if we get back to this arachidonic acid here disrupts the membrane. Any disruptions will cause an increase in fluidity. Certain ion channels don't work that well. They're kind of sluggish in some people. And there is a hypothesis, again, not proven, a hypothesis, certain forms of mental illness, psychiatric disorders, depression, bipolar disorder, one that I'm familiar with. We do a lot of work on that in the lab, and I've got some relatives who actually have bipolar disorder. They think that the ion channels aren't working properly. A lot of neurotransmitters act through ion channels. And if you can change the fluidity of the membrane, these ion channels might work better, and you'd have a treatment. This is where the idea of taking fish oils, which are omega-3 fatty acids, very often polyunsaturated fatty acid, PUFAs, polyunsaturated fatty acids, PUFA, PUFAs. And the omega-3, that means from the end. So there's different ways of numbering these. That would be 3 from the end here. Omega, alpha is the beginning, omega is the end. Omega-3 is 3, and you get a double bond. Fish oils, so deep water, cold water fish. That's where they say what you should eat, cold water fish. From everything I told you now, you should be able to understand, cold water fish, to keep their membranes fluid, they probably have a lot of polyunsaturated fatty acids. That's true. That's why we get cod liver oil. It's nasty stuff, but it has a lot of polyunsaturated fatty acids. So people are taking this for dyslexia, depression, bipolar, even dyslexia. He's emeritus now, but John Stein, the brother of the television food chef Rick Stein, is in Oxford at Magdalen, and he promoted, along with his brother, fish oils for dyslexia and other ailments. So there might be some mechanistic basis for this. The jury's still out. A lot of these things, you get people who are emotionally linked to them, and they have their opinions, and they don't use evidence to sway them. They're interesting, fascinating ideas. The evidence isn't compelling yet, as far as I'm concerned. You might look at it and think the evidence is compelling one way or another. Interesting stuff, all due to membrane fluidity. So getting back to membranes as a barrier, here I've depicted a membrane showing hydrophilic and hydrophobic groups using this little circle and the two legs. It's the hydrophobic core of a membrane that prevents molecules from passively crossing. If something's too polar, they won't cross. It won't cross a membrane unless there's an ion channel or a transporter. Hydrophobic things, non-polar things can cross right through. Unfortunately, a lot of toxins are quite hydrophobic. Probably evolution drove them towards that. Industrial chemicals that are dangerous, very often they're very hydrophobic. When I give you a lecture on drug metabolism and pharmacokinetics, we'll talk about some compounds that are very dangerous because they're hydrophobic. If they weren't so hydrophobic, they would never get into you, and they wouldn't be a problem. So let's now look at molecular details of this. So throughout this course, what I want you to do is look at the molecular structure of molecules and think about what are the physico-chemical properties on that molecule, and how does it impinge, how does it impact on biology? That's pharmacology in a nutshell. How does chemistry affect biology? What we're looking at here, this is from that book, Molecular Biology of the Cell. So go and read that if you are interested in this sort of thing or you want to learn more about it. This is our bilayer. So each one of these bands is a monolayer. It comes together as a bilayer. Small molecules, The thing that dictates whether a molecule can pass or not is hydrophobicity mainly, but it also is size. So small hydrophobic things easily move through. Large hydrophobic things will still move through. Hydrophobicity, therefore, I think is more important than size, but size also has an effect. Here we've got oxygen, carbon dioxide, nitrogen. So diatomic and triatomic molecules. These oxygen doesn't have any dipole, so it's hydrophobic. Not that it can't share, it doesn't have a dipole, it can't share electrons with water. It just passes through membranes. We are the evolutionary product of that because we use oxygen to get a lot more energy out of burning hydrocarbons, fats, or carbohydrates using oxygen than we could without. Carbon dioxide, it's what we burn these two, water and carbon dioxide. So carbon dioxide freely transports. It's hydrated ion, though, carbonic acid. It will form carbonic acid, then dissociate, and you'll just get carbonate floating around in the water. That will not cross, and there's transporters for that. There's also enzymes that help to form carbonic acid. They're called carbonic anhydrases. Nitrogen will pass through. That's neither here nor there unless you're a very deep diver and you come up too fast, you'll get the bends because nitrogen will come out of solution. Benzene, so this is the most basic aromatic compound, 6 carbons. It will easily cross through membranes. It will, don't want to get too much into it, but it partitions between DNA bases, and these are called intercalators, and it can end up causing cancer. You'll get misreading when you replicate the DNA, and it can cause mutations resulting in cancer. Some people in the past have said, which one of these? Oh, CO2. They said, but carbon and oxygen have different electronegativities. Yes, they do, but if you remember, the structure of CO2 is linear. So even though they have a dipole, the dipoles cancel. So it still easily passes through membranes. Water, glycerol, ethanol, small. They're polar, but they're small. So if you're polar, sometimes small can compensate for being polar. Weirdly, very, very weirdly, there's no consensus, at least, on how water crosses membranes, which is kind of shocking. There's like 4 theories going. Each group, each cohort, each school of thought thinks they're right and the other ones are wrong, and they throw mud at each other. So it's really interesting if you're into that sort of thing. Intellectual debates concerning how water crosses membranes. Fundamentally important. And it's shocking to me that we don't know for sure. There's one theory that I think is really, really cool. It's probably wrong. Most of these things that are really, really cool are probably wrong. Nobody has evidence. But what they showed was a diagram where even if you have something like stearic acid, 18 carbons all saturated, you can get conformational changes, this eclipsed and Gaussian whatever that is, rotation around single bonds. They'll form a kink, even though it's all saturated, it'll form a kink in the bond. Water fits in that kink. And then if you run the kink all the way down from one carbon to the next, to the next, to the next, you form this screw. So it's like Archimedes' screw. Archimedes was the guy that jumped in a bath and the water overflowed. He ran out into the streets screaming Eureka, and he then knew how to determine if the emperor's crown was pure gold or a gold silver mix, and the smith who made it for him cheated him or not. Archimedes did a lot of things. One is a screw. It's used for pumping water. It's used for augering grain, that type of thing. It might be how water crosses membranes. Interesting idea, not a lot of evidence. Nobody really cares about it. Nobody really will give funding for this, but it's fundamentally important, and I think it's very cool. The flip side of that is some people say you need pores, you need proteins that form water channels. There certainly are. There's a group of proteins called aquaporins. They're important in the kidney. Are they important in other things? It's difficult to know. There's all sorts of theories swirling around water. There's a really good book just called H2O written by the former editor of Nature, and I would suggest you give that a read. Water has so many weird scientific theories around it that were proven wrong, but you still have these conspiracy theory types that they won't accept any evidence. And there's like poly water, and there's like homeopathy, memory water, and different states of ice. It's fascinating. Go and read that book. Philip Ball, I think, was the author on that. So then we move down to amino acids. So amino acids aren't that big. The average size of an amino acid is about 110, 120 Daltons. But the amino group and the acid group, carboxylic acid group, are charged at physiological pH, so they will not cross membranes. The reason the charge becomes important is it will form a tight shell of hydration. So there will be water molecules lined up in one shell, looser in the second shell. To cross a membrane, you have to strip all that off, shove a charge through, and then rehydrate. Just too energetically costly. Then down here, we have full-on ions, same thing. These are inorganic ions, very small point charges, meaning that they will have even a tighter shell of water, hard to strip that off and get them across. So that's sort of the take home from this slide is in the title. Crossing a lipid bilayer, it's that hydrophobic core, depends on size and hydrophobicity. To the right of this, in molecular biology of the cell, they put numbers to these things. So the numbers go from 10 to the minus 12 to 10 to the minus 2. So those are just numbers over 10 orders of magnitude. That's kind of interesting, but is it neither here nor there? What are these numbers? This is a permeability coefficient. So if you know what the concentration is on one side of the membrane, you multiply it by this permeability coefficient, and you can calculate how fast, what the flux of molecules per unit time is across that membrane. The units here are in centimeters per second. And initially you think, what the hell is like 10 to the minus 2 centimeters per second? Again, like Feynman says, think about this in simpler terms. What's centimeter per second? That's distance divided by time. Distance by time is speed. This is just a measure of speed. We happen to be using weird units. Centimeters per second is just what people in this field initially used for reasons I don't even remember if I know right now, but we're not going to get into that. The other thing, thinking like Feynman, let's see if we can put this into something that we would know about, terms that we could relate to. So here, 10 to the minus 2 to 10 to the minus 12, speed. Let's think about how fast does somebody walk? They're a fast walker, 3 meters per second. Then let's do, instead of 10 orders of magnitude here, or here, if we go, instead of doing 10 orders of magnitude, let's do 8. Is there any number that you know of that's 3 times 10 to the 8 meters per second? Speed of light. So the difference on this scale goes from walking speed, down here, the chloride, up to the speed of light. That's how big of a difference we're looking at here, and how fast different chemicals cross membranes. So it's a huge variation. We can deconstruct this a little more. So flux, any type of flux, so you could do pressure, fluid moving through a pipe, anything like this. Ohm's law is basically this. Electron movement is the voltage divided by resistance. Here we're looking at molecules crossing a membrane. To the right here, I've made a diagram of, this is of a lipid bilayer. Our concentration outside the cell is high, concentration inside the cell is low. And I express that as DC, so delta C. That's our concentration gradient. The flux, that will be the driving force, is our concentration gradient. Resistance, what's going to stop things from going through? The hydrophobic core. And also a little bit this diffusion coefficient. So most small molecules that are drugs are all kind of around this 500 Dalton size. So we don't see huge differences in diffusion coefficients for crossing membranes. As soon as we start talking about messenger RNA for like Moderna or BioNTech's vaccine for COVID, these are big molecules. All I'm saying here goes out the window. You got to use a different approximation. For small molecules, this works. All the diffusion coefficients will be about the same. So we've got the same width of a membrane. It's about 0.3, 0.4, depending on if you just take the of a nanometer, if you just take the hydrophobic core or with the head groups, it's on those sort of orders of magnitude, that type of size. But the diffusion coefficient's about the same, the membrane's about the same, concentration will change. But I said, what dictates the concentration in the membrane, what will cross, or what dictates whether something crosses a membrane or not, is that hydrophobic core. We can do a calculation, we can make a measurement about how well a molecule will dissolve into something hydrophobic. And that directly correlates with how fast it crosses a membrane. So here's our overall fix equation. Here's our flux is diffusion coefficient times the concentration gradient over dx. We can also look at flux in terms of this PM. So we can set a concentration, and then we can calculate the coefficient, this permeability of the membrane coefficient in this centimeters per second. Taking all of this together, permeability coefficient, our diffusion and distance across the membrane are the same. So permeability coefficient should scale directly with log P. Log P is a measure of the greasiness, the hydrophobicity of a molecule. And indeed, that is the case. Down here, people do plots. This is just sketch. This is just my cartoon. But you can go on the internet and look up papers on this, and you'll see over 5 orders of magnitude, these two scale together very nicely. So this is permeability coefficient, how fast it's going across the membrane, against log P, how greasy it is. So the greasier it gets, the faster it'll go across. The way they do this traditionally was a so-called shake method. You put your drug into a solution of water and octanol. Octanol's got 8 carbons of hydroxyl. It's not miscible with water, so you'll get 2 phase separations. So you shake the hell out of that, wait for the phases to separate, measure the amount of your drug in the octanol versus the water. If you've got 10, let's say 100,000 molecules of drug in the octanol compared to every one in the water, that's a 1 to 100,000, 10 to the power of 5, log of 10 to the power of 5 is 5, so that would be a log P of 5. If conversely, for every 1 molecule you had in octanol, you had 10 in the water at equilibrium, that would be 1 divided by 10, 0.1, so that would be a log of -1. So that would be very hydrophilic compound. In general, drugs, you want them between probably log P of 0 and 5. That's the Goldilocks region with this. If they go higher than log P of 5, you won't get any dissolved in the aqueous. It'll just stay a greasy spot somewhere, and you won't get any into the aqueous phase. If you go less than log P of 1, you guys should know what that means. It's too polar. It's not going to cross this inner bit of the membrane here where there's that hydrophobic barrier. This concept forms one of the ideas in so-called Lipinski's rule of 5. Chris Lipinski worked for the drug company Pfizer in Sandwich in the UK. They used to have a big institute down there. Your generation probably doesn't even remember that. There were thousands of people working for Pfizer in Sandwich. This whole place shut. It was devastating to British drug discovery, unfortunately, and development. Anyway, Chris Lipinski worked for them. They were doing combinatorial chemistry back in the day, where they were doing high throughput screening. I'll tell you more about this in my drug discovery lecture, but basically they were just making compounds, and a lot of them were failing in the assays. So he said, can we look at physiochemical properties to see why they're failing? And one of them was too high of a log P. The compounds were too greasy. They wouldn't go into solution to cross membrane. So he said log P less than 5. Then he also looked at hydrogen bonding. You wanted compounds to be a good drug. It should be less than 5 hydrogen bond donors or less than 10 hydrogen bond acceptors. And it also should be less than 500 Daltons. So 500 Daltons relates to size. I said for things to cross membrane, it depends on size. Hydrophobicity, that's a log P less than 5. Hydrogen bonding relates to whether something's polar or not. So it also relates to whether these things cross membranes easily or not. So you'll hear the rule of 5. It's not that there's 5 items to the rule of 5, there's only 4. The rule of 5 means that everything in the rule of 5 is 5 or divisible by 5. So you'll hear Lipinski's rule of 5. It all relates to whether these molecules can cross membranes. So that's a very useful strategy in an early drug development program. You can screen out a lot of molecules that wouldn't be bioavailable. Not all molecules cross membranes passively, though. Some of them will be transported across a biological membrane through these carriers that I've talked about. These are integral membrane proteins that go across the membrane. As long as the membrane's in this fluid mosaic state, they can function, they can swell and collapse, conformational changes. They'll take things through like glucose. So that's one way to get some of these polar but big compounds through. Ions, inorganic ions, we have ion channels. These open up and they allow ions through, but they have selectivity filters. Sodium channels will be, let's say, 1,000-fold more selective over potassium. They also don't let water through. We have specific water channels that I told you about earlier. So some of these things, they need pumps or channels to get across. So the membrane is a physical barrier. We call it selectively permeable. That's because of these proteins in the barrier. If you just had a pure lipid bilayer without any of these biological proteins in it, only passively permeable compounds could get through. The other thing that's interesting from my standpoint to think about is that these membranes are not just barriers to matter, to molecules. They're also barriers to signals. Something like adrenaline can't cross the membrane. Its receptor is in the plasma membrane, and the signal of it binding gets converted into other chemical signals in the cell. This is a so-called first messenger. Adrenaline, if it's a G protein coupled receptor, will be converted into what's called a second messenger. It's a whole concept from adrenaline down to an endpoint in your liver where it releases glucose from glycogen through an enzyme called phosphorylase. There's a bunch of steps. These are molecular dominoes that just keep falling onto one another, going from adrenaline down to the enzyme that releases glucose. 6 Nobel Prizes in that pathway. So this is important. Back in the day, it was very novel stuff. It forms the framework of how we think about cell signaling now. So membranes can be barriers to matter, to molecules and ions, but they can also be barriers to information. There's also something that you might want to think about and go and read about. There's this interrelationship between matter and energy, Einstein's equation, of course, everybody knows that. There's also a relationship between signals, information, and thermodynamics. Entropy is information. So cells are like anti-entropy machines, and signaling across plasma membranes can be related to some of these energy effects. And keeping a cell intact is an anti-entropy thing. Life in general, our being here, is anti-entropy. There's some interesting ideas about that. Read about Maxwell's demon, read about or watch some videos by a guy called Leonard Susskind or Susskind. He's actually a physicist, came up with string theory, also a holographic view of the universe. The whole universe could be on 2 dimensions. It's really cool. I don't want to go down that route too far. But if you're interested in how information can be equivalent to energy, watch some of that. Leonard Susskind, really cool stuff. So from this, we've got a barrier to matter and information, and these integral membrane proteins help us to breach that barrier. So to give you just sort of a feeling for this, a rule of thumb, and in biology, there's always spectrums of differences. Here, there are some molecules that can cross through directly. They're hydrophobic enough. They're in that Goldilocks region of this log P between 1 and 5. They're soluble enough that they can be in the aqueous environment, but hydrophobic enough that they can cross membranes. Steroid hormones, based on the cholesterol nucleus or the steroid nucleus, these things just slip through. They don't need any carriers. Ion channels. Very often biologists, when we say ions, usually we mean inorganic ions: sodium, potassium, chloride, calcium, magnesium. Other organic molecules might be in ionized states, but very often we don't call them ions as such. We will say carboxylate, and it is an ion, but we don't necessarily address it as such. So ion channels, these are like 10 to the 8 per second. An example would be sodium. The small molecules that need transporters, these can either be passive. So when your blood glucose is really high, there'll be a glucose transporter that just takes the high concentration of glucose in your blood into cells by a revolving door mechanism. If the cell happens to get a really high concentration, it will spit out glucose into your blood. That's what your liver does when it degrades this glucose polymer called glycogen. If you do the fight or flight response, you get scared or you're competing in some athletic event, adrenaline increases, it goes through all those molecular dominoes, releases glucose into the blood. That happens in the liver. So some of these, whatever the gradient is, it just helps them achieve that gradient. We also have what's called active carriers. These rely on a sodium gradient. There's sodium gradient across membranes. Sodium's 140 millimolar roughly outside cells, and roughly, what is it? Forgetting this now, I think 14 millimolar inside cells. So that gradient, other molecules can use it. So if, again, using my revolving door analogy, if in the one, if you're glucose, you're pushing your own revolving door. If it's an active transporter, sodium's in there giving you a big push. So it can actually crowd more glucose into a cell. It can actually concentrate things into cells. Also ATP, that energy molecule I mentioned earlier, that results from mitochondrial respiration, that some molecules are powered by cycles of phosphorylation and dephosphorylation, and it flips it inside and outside. And that can also cause concentration gradients, especially with sodium potassium ATPase. That's what establishes a sodium gradient, kicks sodium out, potassium in, and then that sodium can be used by other things. These are called co-transporters. An example of this would be adrenaline, and an example of the ATP would be sodium potassium ATPase. Look at the speeds here, 10 to the 8, 10 to the 3, 10 per second. Again, think like Feynman, think about what that might mean. Channels are fast, pumps are slow. That's the elevator pitch, the Twitter feed on that one. So we've been talking about membranes and barriers, and the barrier is mostly that hydrophobic portion inside the membrane. Membranes are just fit together by thermodynamic forces, mostly hydrophobic forces, where hydrophobic tails are just excluded from the aqueous phase. That's the driving force for the hydrophobic force. Now I'd like to extend this a little bit into something called the blood-brain barrier. So again, this uses cells, but it was discovered by, and it's important in keeping toxins, xenobiotics. If we eat things, the body doesn't want to poison the CNS. That's really bad news. So I'll give you a lecture later in the term on drug metabolism and pharmacokinetics, where we'll talk about how the body goes about detoxifying drugs in a two-step process to urinate them out or defecate them out. But nature has evolved protection around the central nervous system. The central nervous system means the brain and the spinal column. So the brain, the brainstem, and the spinal column is our central nervous system. And this is a very privileged structure that you don't want to get wrong. In terms of this Paul Ehrlich, some of you might know him, some of you might not. He got a Nobel Prize in 1908. We're talking in this whole lecture about targets. I haven't really defined a target yet. I will in a minute. But it's intuitively obvious. Ehrlich was playing around with dyes. There's a dye down at the bottom here called trypan blue. These dyes he would add to biological samples, microbes very often, trypanosomes, that's where this trypan blue comes from. And it would stain certain parts of the cell, but not other parts of the cell. So he came up with this idea, and he published the idea of a magic bullet. He didn't say magic bullet. He was German. There's one German word for magic bullet. Of course, it's one word. They're Germans. Can't remember what that is off the top of my head, but there's this magic bullet. If you have a magic bullet, bullet has to hit something, hits a target. So Ehrlich had the idea of magic bullet and target. He also coined the term antigen and antibody, which is all the rage with COVID right now. Some of you might be doing lateral flow tests. You're either looking for the antigen or antibody lateral flow or PCR. So he was instrumental in our ideas about chemotherapy. He said if we had a magic bullet, meaning it would hit a parasite and not our cells, that would be ideal. That's chemotherapy in a nutshell. And that's what the whole field of pharmacology has emerged from. Ehrlich also experimented with arsenical compounds. The very first drug, salvarsan, was an arsenic-containing compound, treated syphilis. So he did some of the very first effective chemotherapy where it wasn't just using natural products without any real underlying understanding about what was going on. So Ehrlich, Nobel Prize, magic bullet and targets. Injected a rat here with trypan blue. And notice the rat's ears and its tail and its paws and its eyes and nose are all blue. But when he cut that rat open, he doesn't show this in his paper, but he did show in his paper this one. This one is a chick embryo. This chick embryo, so it's a chick, if you aren't aware of this chick would be like sideways, lying on its back, cut in half, and then visualized. So this is a section through a chick. Everything's blue except the brain and the central and the spinal cord. So this trypan blue was excluded. Now I want you to have a look. If we were in class, we'd play this game. Tell me about this molecule. Tell me why you think it might be excluded. Hopefully you'd hone in on these SO3 groups. Physiological pH. pH in biology is always going to be around 7. There's some exceptions, usually around 7. Those are all going to be ionized. The pKa on that's probably around 1 or 2. I can't remember off the top of my head. So it's going to be ionized at physiological pH. It's also a fairly big molecule. We've got a lot of structure here. So we've got a big molecule and a hydrophilic, a polar molecule. This is not going to cross membranes, but when they injected it, it went everywhere, except the blood, across the blood-brain barrier, what has come to be known as the blood-brain barrier. It went everywhere because some cells, even though crossing into cells, you have to go through a membrane, not all the cells have good contacts between them. They're leaky between them, most of them. So that's why it was able to go into all these spaces. The exception is the blood-brain barrier. Here we have a group of cells that have what are called tight junctions. They come together very tightly. Molecules can't get through those. So this was the first demonstration of the blood-brain barrier. And it's important for central nervous system drugs. Interestingly, just go back for a second, if you just based everything on theory in biology, you would make some grave mistakes. So if I showed you the structure of several antibiotics, you'd say they're charged, they're big. If somebody had meningitis, which is an infection of membranes in the brain, so this is a bacterial infection of the membranes in the brain, you would say, don't give them antibiotics, you'd be wasting your time because they wouldn't cross the blood-brain barrier. That's the theory behind it. Happily, nobody really paid attention to that. Medical doctors just gave patients with meningitis antibiotics and they worked. They worked because when you get a bacterial infection in the meninges, the blood-brain barrier breaks down. There are other ways of breaking down transiently the blood-brain barrier. They're trying to use this to get treatments into the brain. At the very end of this lecture, I'll tell you a little bit about antibodies. Antibodies are really big. They're proteins. They're like, I can't remember, 75,000 or 175,000 Daltons. They're not crossing a membrane passively. So how do you get those into the brain? There are theories around that, but it relates. This blood-brain barrier is an important thing. My little anecdote about biology is, doesn't matter what you think theoretically, do the experiment. If it sounds really, really stupid and you don't want to tell anybody and you're embarrassed, but it might be a really cool thing if it works. These are like Friday afternoon experiments where everybody else has gone down to the pub. You stay behind, do the experiment. If it doesn't work, you don't have to embarrass yourself in front of anybody. If it works, you're a hero. A lot of big breakthroughs occurred through these sort of, in quotes, air quotes, Friday afternoon experiments. In terms of the blood-brain barrier, this is a cross-section, a cartoon cross-section through brain tissue. Out here, there are neurons and glial cells. Neurons are the ones that depolarize and send neurotransmitter and talk. They're the electrically excitable tissues. It's probably where consciousness and higher brain function and all that occurs. Glial cells are equal in number, perhaps maybe even higher in number. The name glia comes from Greek, I think, and it means glue. They thought this was just the mortar and the neurons were the bricks. We now know that they play a more active role. They're helping all the neurons all the time with neurotransmitters, potassium toxicity, even metabolism of glucose. They metabolize glucose to lactic acid, lactate, and then give lactate to the neurons, which the neurons then burn in the mitochondria using oxygen. So they're important cells. Anyway, everything outside here is neurons or glial cells. In the middle here, this is blood. So this is a capillary, very, very fine. So your circulatory system keeps dividing, till you get down to where there's like, here it depicts 1, 2 endothelial cells, endothelial. So epithelial is on the surface, endothelial means internal. It's the internal surface of these blood vessels. And in other tissues, these would be leaky. You would have big spaces between the cells, but in blood-brain barrier, there's tight junctions. There's proteins that stitch these together really tightly. Molecules can't pass. Any molecule that leaves the blood has to cross through 2 membranes, plasma membranes, to get into the brain. Plus, it has to cross, I don't know if it's here. No, I don't have it here. But there are pumps, like some of these pumps. These are showing pumps pumping sugars and amino acids into the brain, because those aren't passively permeable. They have to be pumped into the brain. So the brain has carriers to take them through. Molecules you don't want, if they happen to leak into the cell has pumps called the P-glycoprotein or the multi-drug resistance transporter or the breast cancer resistance transporter. And these will take chemotherapeutic drugs and spit them out. That's why they're related to cancer. Evolutionarily, they're there so that when we eat bad plant products, toxins, and if they're hydrophobic enough, they'll leak into the cell. The endothelial cell then will pump it back into the blood, and you'll eventually pee it out. So this is the blood-brain barrier. It's very important. It relates to membrane transport. So now, finally, you're wondering, you thought the lecture was on drug targets. Where the hell are these targets? We're finally there. So I've organized these in terms of Rang and Dale. Rang and Dale is a pharmacology textbook that's popular in Oxford because Maureen Dale was a member of the Department of Pharmacology in Oxford. She and Humphrey Rang did some of the initial experiments with making radioactive compounds and showing receptors. I'll tell you about that in the next lecture. They wrote this textbook called Pharmacology, so we use it here. Everybody here thumps their chest and says it's the best. It is a textbook. It's kind of a ridiculous game saying which is the best textbook and which is the best this and that. It's a textbook, and read that one, read other ones, see which ones you like, find one that suits your personality and just go with that. Rang and Dale divide drug targets into 4 things. Then I'll also divide receptors into 4 subgroups. So I'll walk through these. Don't have time to go into detail on all of them, but what I'll try to do is give one very specific example that gives you a flavor of that particular drug target class. And then when you see another one, if I'll tell you about an enzyme, and aspirin. This is the cyclooxygenase that I already foreshadowed. If you see another enzyme, everything I tell you about aspirin and cyclooxygenase should apply to that other enzyme. That's why I'm doing this. So we've got enzymes, carriers, I'll do serotonin, ion channels, dihydropyridines, receptors, I'll do the mu opiate receptor as a G protein coupled receptor. Throughout these, sometimes I write this in, sometimes I just try to remember to tell you. As an aside, all these targets are based on small molecules. It's becoming a brave new world. There are drug approvals now by the FDA, the Federal Food and Drug Administration in the States, and the EMA, European Medicines Agency, of siRNA, so things that inhibit the expression of messenger RNA. They're doing this for Huntington's disease. The data look fantastic. How the hell these things get across membranes, and like the messenger RNA vaccines, it's a bit of a black box mystery. And I used to be very against these. I'll say, I didn't think that I thought they were insurmountable challenges. I was very wrong. People are getting around those, and I'll tell you more about that when I give the drug discovery lecture. But we're going to have to, we as a society, scientific community, have to start thinking about different types of targets rather than just these old ones. There's also something called protein condensates. These are regions of the cell which don't have a membrane-bound organelle. So organelle means mini organ. That's the way we thought about organelles. There are regions of the cell for various reasons at temperatures or signaling patterns that seem to come out of solution and form oily drops. And then they'll go back into solution. Again, that seems to relate to Huntington's disease. The disease is ON at the end. The protein that goes awry in that is Huntingtin, IN at the end. And Huntington has this polyglutamine, an amino acid glutamine at the end. As that gets longer and longer and longer, you eventually start getting neurological disorder. And it's a death sentence, and we can check your DNA and see which variant you have and basically tell you what time in your life you should get your things in order based on how long this is. Nobody really understood it, but these polyglutamines seem to be forming these condensates, these oily bits. We don't fully understand what's going on. Some DNA, some RNA seems to be forming these as well. There's something new under the sun here, and it's really exciting. Don't know what it means yet. It might just be an artifact. We don't know, but because this Huntington protein's involved, that suggests that it might have something to do with disease and possible treatment of the disease. So that's just something for you guys to think about. In your careers over the next 50, 60 years, you'll see some of this come on, or it falls by the wayside. It was just a dumb idea, but we don't know if it's a dumb idea or not. Like I say, biology is an experimental science. We need to do the experiments. This one, I was going to include more. I almost said everything that I was going to say. I was looking for this and I couldn't find it. Then I found it, but it's too late for me to put it in. There's a paper in the journal Nature a couple of weeks ago, a review on vaccinations by Andy Pollard, who's head of the Oxford Vaccine Group. Look at that paper in Nature. Look at their graphs over time with deaths, and they show an inflection point where the vaccine was introduced. If you have any friends, or even if you have doubts in your own mind about vaccinations, look at that. It's unequivocal evidence. I don't see how anybody can be anti-vax after seeing some of that type of stuff. We as a society, this is science to our advantage. We're making great progress. Doesn't mean that all vaccines are completely free of risk. If you go outside, even now, you can still get run over by a car or something. Life's not without risk, but vaccines are incredibly safe for the benefit. There's always this risk-benefit analysis. Membrane, again, think about how these messenger RNAs might cross the membrane. There are ways in which they did it. I'm not going to tell you. If you're interested, go and look them up. Very clever. The basic science that went into those messenger RNA vaccines was in danger of not being funded, and people were going to lose their careers and have to go on and do something else until the pandemic. And now it's revolutionized how we go on in the future making vaccines, probably. It's tremendous. At no point in history could we have ever made vaccines this fast. It used to take us about 15, 20 years. Some things we still don't have good vaccines for. This is incredible, and it's built on the basis of basic science. So our first drug target class is enzymes, according to Rang and Dale. This is arachidonic acid, which I talked about earlier. For pure chemists out there, don't get too bothered. We non-chemists who dabble in biology, biologists who dabble in chemistry, maybe that's a better way of phrasing it. If this is uncharged, we know it's arachidonate. If it's charged, it's arachidonate. If it's protonated, as the product I show over here with an acid, that would be arachidonic acid if it was on the other one. Biology is always around pH 7, so we don't control what charge is on ionizable groups. The biology does it by dictating pH 7. So arachidonic, arachidonic acid, they're the same to a pharmacologist or a biochemist. To chemists, if you're trying to get some compound into your organic phase from the aqueous phase, it's damn important, and you can control that. In biology, we can't control this, so we're more lax about that terminology. A good example would be DNA, deoxyribonucleic acid. All the phosphates on the backbone of that will have negative charge at physiological pH, but we don't call it deoxyribonucleate. We don't worry so much about the acid-base stuff. We just, it's a compound, and the biology will dictate that. So our friend cyclooxygenase that I told you about, it will introduce oxygen here and form a peroxide. This is a short-lived product. It actually introduces two oxygens, one up here, one down here, but this peroxide is even more unstable, and it switches quite quickly. Prostaglandins, you can look them up on Wikipedia. There's like a whole herd of these, and they all are structurally different. They all have names. They're all important to people who work on them. For us, we're not going to worry about that. This is a short-lived intermediate, but I think it's interesting because it shows you what cyclooxygenase does. I think it's prostaglandin G, and then one of the ones that gets released is GH, and then GH2, and all of this. You can go and look it up. This enzyme also will make substrates for arachidonic acid, will go to thromboxane, which is a whole nother group of signaling molecules. And it also makes leukotrienes, I believe. Sorry, no, not leukotrienes. That's a different pathway. It will make something cyclins. It's a 6-member ring. Sorry, I think about all of this in terms of structures. I could draw it for you. Prostacyclin. I could draw it for you, but it's hard for me to remember the names all the time. I'm a bit dyslexic, so it's a challenge converting the images in my head to words and actually written words as well. That's why all my slides are pretty word-free, minimalist. They've got images. The images is the way I think about this stuff. For you guys, you might want to annotate this or rewatch it or that sort of thing to make sure you get all the information from this. So prostaglandins, these do pain, inflammation, and fever. Enzymes that inhibit cyclooxygenase, John Vane got a Nobel Prize for that. One of them was aspirin. Aspirin's an interesting compound. It inhibits cyclooxygenase. What we found then, though, after Vane's discovery, was that there's multiple forms of this. As I said earlier, all of these targets seem to come in families. There's isoforms of them. So the isoform 1 is housekeeping. That's in all our cells. It keeps things ticking along. In the stomach, if you give somebody aspirin, it inhibits cyclooxygenase 1, and that reduces prostaglandins. Prostaglandins in the stomach are necessary for signaling to release mucus and bicarbonate so that the acid released in the stomach doesn't hurt the stomach, the epithelial cells in the stomach per se. The stomach will get so acid it'll get to pH 1, so 0.1 molar HCl is effectively what stomach acid is. When you have the unpleasant experience of vomiting, you know that stomach juices are very acid. So aspirin will inhibit this protection mechanism, and people will get bleeding ulcers, and aspirin kills probably about 200 people in the UK a year. Contrast that with probably the people that die from MDMA, ecstasy. It's probably between 10 and 20 a year, arguably. So it's a bigger problem if you just look at death rates, but it doesn't make the front page of the Mail when somebody dies from a stomach bleed from aspirin, because usually it's an older person trying to treat their rheumatoid arthritis, this sort of thing. Then they discovered cyclooxygenase type 2. As soon as you have two isoforms, you can start thinking, can we target one selectively from the other? What's interesting is, rather than housekeeping, COX-2 is pain and inflammation. So if you slam your finger in a car door, you'll get all swollen up and be very painful. That's because of this cyclooxygenase type 2 being induced. Induction of an enzyme means it goes from DNA to RNA, there's transcription, and then translation into protein. So the enzyme normally wouldn't be there. It gets upregulated 100 to 200 times upon injury. Companies then made inhibitors for type 2. Type 2 has a little pocket on the side. Type 1, there is a leucine that pokes out. Leucine's a little longer than valine. Valine, I do this because valine is V and it actually has that type of structure. It protected this pocket with isoleucine. In the COX-2 one, it was isoleucines in the COX-1, no access to the pocket. In type 2, valine's there, one CH3 unit less, the pocket was accessible. So they made rofecoxib, you can see here, with a side chain. And that side group, that pokes into this hydrophobic cavity. So you got selectivity over type 2 over type 1. This revolutionized the treatment of rheumatoid arthritis, which is crippling, debilitating. Somebody would have to spend their days in bed. It mangles the joints in your fingers so you can't move them. So mechanically you can't move them, plus it's incredibly painful. It's really debilitating. The only thing we used to have is Nsaids, these non-steroidal anti-inflammatories, and aspirin you can't take at high doses because it causes these stomach bleed problems. So rofecoxib, rofecoxib is the generic name. Then the brand name from Merck & Company was Vioxx. They were making billions of dollars a year on Vioxx. And then some GP in the States noticed and publicized that he was seeing more cardiovascular events with people on Vioxx. There's a yellow card system where if there are untoward drug reactions, doctors can send a yellow card in to the regulatory agency. And then if they get a bunch of yellow cards saying there's cardiovascular events with this, they can put a hold on this, they can investigate. So yellow carded this. And it's true, there were events, cardiovascular events, mostly clotting problems, thrombosis. A thrombi is a clot that you don't want. It moves around your circulation, gets jammed in one of these positions where your vasculature is splitting down and getting narrower and narrower. Then you have thrombosis, you can have a stroke. It's all bad news. You stop perfusing tissue with oxygenated blood. So this happened. Merck got sued. They withdrew their product. It was the biggest class action lawsuit in history at that point. Every pharmaceutical company had COX-2 inhibitors. That's why they like to know what the drug target is, because then you can make inhibitors of it. And they all killed their programs, and you can't get highly selective COX-2 inhibitors. There are some. Celebrex, I think, is still being sold in the States, and it's maybe 2-fold to 5-fold selective. Rofecoxib was like 100-fold selective for aspirin. Turns out that prostaglandins are involved not just in pain and inflammation, but platelets, the cells in your blood that stick together or not. If you're wounded, you want them to stick together. If you're not wounded, you don't want them to clot. So it's on a knife edge. It's walking a tightrope, whether you respond too early and you get a thrombosis, or you don't respond at all and the person bleeds out. So it's a very important regulation. And if you inhibit COX-1, it doesn't affect this seesaw balance in this. If you inhibit COX-2, it tips it towards aggregation of platelets, towards clotting, and that was the problem. I happen to think it should still be out there. A personal aside, I had a great aunt in Canada who was in her 90s, and she had rheumatoid arthritis, and she was bedridden. She happened to be a Roman Catholic nun, and she lived in where kind of, it wasn't really a convent, but a place where with a lot of other nuns attached to a church, and she couldn't get out of bed and go down to the church to pray. My brain does not have that bit of sort of religiosity, spiritualism. I think we're nothing but chemicals, and that can explain everything. It's just the way I think, and I blame it on my brain, that sort of thing. But hey, if my great aunt thought her life depended on having the life of religion that she wanted, and the movement to being able to go down to the church to pray, it was given to her with Vioxx for a short period of time. Then it was withdrawn, and she was effectively bedridden again. I think somebody in their 90s should have the choice with their in consultation with their doctor to decide whether the cost benefit is there, what they want. I don't think she would have minded if she had a cardiovascular event at that point in her life, but she really desired to be able to get mobile and go and talk to people, go down to the church and things. So I think it was a mistake by regulators. It's still controversial. There's various people. The pro-vice chancellor for innovation at Oxford, Chaz Bountra, used to work at GSK, GlaxoSmithKline. He used to give a lecture for this, but now he's too busy with other things. But he told me stories about that, and he thinks also that rofecoxib and these COX-2 inhibitors still should be out there. And he's debated the American GP in public on this several times. Anyway, shows that these molecules can have widespread implications in terms of society, in terms of lawsuits. All these companies withdrew their drugs because of threats of lawsuit at that. And I think it's taken away one tool that we could use to treat rheumatoid arthritis, unfortunately. Anyway, I'll get off that soapbox. Last thing I'll say here is COX-3. I used to have this in all my lectures. Now, a couple of years ago, I had to put mistaken over it. This is a story of how science works. Nobody knew how paracetamol acted. That's the drug down here. So a lot of people will tell you you have to know the mechanism of action of drugs. You don't. Humanity took opiates. Humanity took cannabis products. Humanity chewed on willow bark, which had salicin, salicylic acid, this part, not the acetyl bit, but just this part. Humanity took all these plant products before we had any idea about receptors, let alone mechanism of action. So I think it's a nonsense. Regulators also, FDA, EMA, do not require mechanism of action. I'll say more about that, a lot more about that when I do the drug discovery lecture. Anyway, nobody knows how paracetamol acts. A few groups do think they know, but they don't agree. So if there's no consensus, I think we as a society of community of scientists don't know. A few years ago then, COX-3 was proposed. So they found a new cyclooxygenase isozyme, and it showed really beautiful dose response curves that fit with the dose. Problem was, it was in dog. They got a really nice paper in the Proceedings of the National Academy of Sciences, one of the glamour journals. And if you're a dog, paracetamol is your friend. If you've got a headache, it will work. When they did the studies in humans, they looked at it genetically, and this is probably 3 to 5 years after that first publication. We humans have a mutation in what would be our COX-3, a stop codon. So every 3 bases codes for an amino acid, and then certain 3 code for a stop codon. We have a stop codon, so we truncate the COX-3. Can't be the explanation in humans. A delightful idea, it's probably wrong, the evidence is really weak, unfortunately, I wish it was strong, is that paracetamol forms a metabolite with arachidonate, and the acetyl group comes off, the amino group bonds here, you get an amide, you get something called anandamide, a Nature paper on that when it came out. Anandamide comes from Sanskrit ananda, meaning bliss. This hits the cannabinoid receptor, and it will give you feelings of euphoria, all the stuff why people use cannabis as a recreational drug. I would like this because the regulators and government don't allow we scientists or companies to work on things that might hit the cannabinoid receptors because of the potential for what they call abuse, the potential for recreational use. I think it would be delicious, delightfully delicious, and ironic if paracetamol reacted with this and that was the mechanism of action through cannabinoid receptors. The data is crappy, unfortunately. I wish it was much better, but it isn't. So anyway, the next target is carriers. So these are the things that can move molecules that wouldn't passively cross a membrane across a membrane. The example I'm using here is for a neurotransmitter called noradrenaline. Adrenaline is produced from a gland just above your kidneys. Adrenaline, adrenal. Renal is Latin for kidney. Ad is above it, so this gland is above your kidney. That's where you get adrenaline from. In the States, they call it epinephrine. Somehow they like to follow the Greek. Kidneys, nephron. Epi means above in Greek, so epinephrine and adrenaline are the same thing, just one's classic Greek, one's Latin. Noradrenaline, nor in chemical nomenclature usually means we know this one structure, but oh my God, it's missing something. We'll name it after that popular structure, but we'll call it nor. Here it's nor because it's missing a methyl group off this nitrogen. If there was a methyl group there, that would be adrenaline. It's not, so it's noradrenaline. There's a related compound called dopamine. It comes from dihydroxyphenylalanine. So this is the dihydroxy. It's also known as a catechol ring. And then that gets metabolized into dopamine. Dopamine, noradrenaline, dopamine, and then there's another one. All of these are amines, so they're neurotransmitter amines. There's one called 5-HT, again derived from an amino acid, the amino acid tryptophan, 5-hydroxytryptophan. It's also known as serotonin. So all of these neurotransmitters, what you're looking at here, they all structure similar to this. You can look them up. They're all amines. So the pKa of this nitrogen, actually, technically the pKa of its conjugate acid, but I just think of a pKa. So pKa of this type of nitrogen, primary amine, would be between 9 and 10. So at pH 7, like only 1 in 1,000 molecules wouldn't be charged. So this is going to be positively charged. That's why it needs the carrier. Neurotransmitters are packaged into vesicles. These are membrane bound. And then these fuse to the plasma membrane when you get a spike in calcium. Calcium causes fusion. This releases the neurotransmitter into what we call a synaptic cleft. Neurons communicate from one neuron to another through neurotransmitters. That was a big deal back in the day. People didn't realize, they thought it was all electrical. So you'd have cytosol connected to cytosol from one neuron to another. And it was just electrical. It's like a wiring of a house or something. But no, there are chemical neurotransmitters. People made all sorts of theoretical arguments, but a guy called Henry Dale provided what arguably would be definitive proof for the chemical basis of neurotransmission. So it's electrical and spit out a chemical, jump the synapse, another neuron fires, electrical and chemical. So it's this jumping along of electrical and chemical signals. These neurotransmitters are released into the synaptic cleft. The synapse is so named from what I understand. In Greek, it means a meeting place. So it's when these neurons come together to meet. Neurotransmitters can then be degraded. Acetylcholine, I'll show you that molecule in a few minutes. It's degraded. There's an acetylcholinesterase, so that cuts off the neurotransmitter. The other way that you can kill a neurotransmission signal is you can either be bad and just chuck it out there and leave it in the environment, the extracellular environment, or you can be more environmentally conscious and recycle it. This is the recycling pathway. It catches a lift on a transporter, takes it back in, then there's transporters actually that take it into this synapse, and you're ready to fire again. there's a limited supply of neurotransmitter in these synapses. So you have a limited amount of neurotransmitter and limited amount of synaptic vesicles. So you can deplete that. Dopamine, 5-HT, and noradrenaline all use transporters. Each of them has their specific transporter. I'm talking in generalities now. This is on the neuron, and it's called uptake 1. It could be on other tissue, and that's called uptake 2. We won't get into those differences, but if you see that in the literature, that's all that means. If you inhibit this, you will leave noradrenaline out there. Noradrenaline's an excitatory neurotransmitter, so that will make people a lot more alert. We know this. Again, it's this theme that runs through pharmacology, how drugs were developed. Cocaine came from the coca plant in South America, and natives there would chew on it for energy when they went on long marches and things. We applied Western medicine techniques to that. We isolated things. We isolated pure white cocaine. Once you get pure white cocaine, you can give it to people, and you can give so much. It inhibits this transporter. You get a lot of noradrenaline out there. People like this, not just noradrenaline. It also inhibits, in fact, a little bit better, dopamine. Dopamine is the reward chemical. So this is the reward chemical, how people get hooked on drugs and why they like things. It's associated with sex, drugs, rock and roll, good food, good living, not just the experience of it, but the anticipation of it. So just thinking, if you are hooked on heroin or cocaine, just the thought of it will give you a dopamine release. You don't even have to go in there and inhibit the thing. Trevor Sharp will tell you a lot more about that in his lecture on recreational drugs. So cocaine inhibits this transporter. You feel really good for a while, but eventually you burn out. You crash. You crash because the noradrenaline stores and the dopamine stores have a finite amount. The cell can't synthesize it fast enough to keep up with it. So that's why cocaine will be addictive, and eventually you'll crash on it. Amphetamines do very, very similar things, with slight differences. Cocaine was considered a wonder drug by a guy all of you will have heard of, Sigmund Freud. So in Vienna, Freud was working with cocaine. He a father of psychoanalysis, father of psychiatry, some might argue. And he, in his writings, you can go and read them, he said it didn't matter what problem people were having, cocaine solved their problem. At least they felt a lot better. So from, mental health ailments to physical ailments, cocaine was the answer. He then found out cocaine was addictive and crashed and all this. Then they started putting people on heroin. Go figure. I'll say more about that aspect of it in one upcoming lecture. Getting back to this, just another interesting aside with cocaine, it's also a local anesthetic. I'll give you guys a lecture on anesthetics, local and general. Freud hung out with a guy who is an ear, nose, throat surgeon called Kohler, can't remember his first name. And they put cocaine in each other's eyes, and their writings, they were amazed that they could poke pins and needles in the whites of their eyes without pain. So cocaine's still used in ear, nose, throat surgery because it not just is a local anesthetic, it releases noradrenaline. Noradrenaline will constrict the vasculature. So smooth muscle wraps around your vasculature, and when it constricts, you won't get blood flowing. So it prolongs the effect of a local anesthetic. My sister's a dentist. The local anesthetic she uses, they put adrenaline or noradrenaline in it, but ear, nose, throat people still use cocaine, from what I understand, for local anesthetic because of that. So cocaine was there, but it made people feel better, but there was this tremendous downside. From that, though, slightly different way and more convoluted story, we ended up getting something called disipramine. This forms a family of tricyclic antidepressants. We now know that these act on, these transporters. Tricyclics hit all three of those transporters. So noradrenaline, dopamine and 5-HT. For depression, we really want to just hit the serotonin one. That's how we got serotonin selective reuptake inhibitors, SSRIs like Prozac, citalopram. Because this tricyclic hit all three, people would get a lot of side effects: dry mouth, dizziness, heart problems, all sorts of things. There was a heavy side effect burden with that. But now that we know the target, people are dialing in now. Instead of just 5-HT inhibitors, they're now dialing in 5-HT and noradrenaline inhibitors as well. Cocaine, if you wanted one for dopamine, cocaine's probably the drug for you because it does hit that one harder than these other ones. So that's carriers. Everything I told you here, you should be able to extrapolate to other carriers. In terms of where this bit is, so this is a really zoomed in cartoon of the N-terminus of a nerve, and that's shown more here. So I used to just leave it the way it was. And then some people said, I don't understand. Nerves all a blob like that? No, that's just a zoomed in bit at the end of one of these synapses here. So this is a cell body, a nerve cell body. Signals come into it. Then there's conduction and action potential you'll hear about. And then there's output here. That's where it releases the neurotransmitter. The input, there will be receptors on the cell surface here, ligand-gated ion channels. I'll tell you more about those in a second. Neurotransmitter binds, it opens up, cations flood in, depolarizes the cell. That depolarization goes to the cell body. Then, depending on the circumstances, it will shoot out down the axon. And then if it hits the end, calcium channels open and you get more neurotransmitter release. You'll hear a lot more about that from Nigel Emtage when he gives a lecture. So our next target is on smooth muscle itself, on smooth muscle of the vasculature. And this is ion channels. These are not ligand-gated ion channels, but what I'm showing you here is a voltage-gated ion channel. So membranes will hold a voltage, and it's usually, give or take, -60, -70 millivolts. You might not think that's much, but take a pad of paper and a cup of coffee and do some calculations. Look at the electrical field across a membrane that's like 0.3 nanometers, and you'll see that this works out to electrical fields like high voltage lines that are hundreds of thousands of volts per kilometer. I can't remember the details, I'd have to rework it out. But there's this electrical field in millivolts across this plasma membrane. If it depolarizes, it loses that charge. Some of these channels, they have a bit in the membrane, like a cork, that moves with the electrical field. So when it depolarizes, the cork pops out and they can conduct electrons. Here I drew it as a gate, just a calcium channel here. We delta, change in voltage, pops open, calcium floods in. Calcium contracts all muscle cells. Doesn't matter if it's skeletal muscle, cardiac muscle, smooth muscle, it contracts it. Smooth muscle is most of the muscle inside us. It's called smooth because it doesn't have striations. If you look at muscle under a microscope, skeletal and heart muscle will have stripes where the fibers that pull on each other line up. You can see those as bands, so striations. Smooth muscle, it's more disorganized, but smooth muscle, it's longer and it will wrap around. So here I show a vein, probably should be an arteriole, but whatever. And there's smooth muscle surrounding this. And here's red blood cells flooding out of that. There's other blood cells in there as well. But when the smooth muscle contracts, the cell body is wrapped around. So when it contracts, the diameter, the radius of this collapses. Smooth muscle is involved in the gastrointestinal tract, in sphincters, peristalsis, movement of the gastrointestinal tract. Smooth muscle is involved in the vasculature. Those are probably the main areas. It's also involved in contraction of the uterus. So during childbirth or during the menstrual cycle, when a woman has her period, the uterus contractions are from the smooth muscle. Like I say, it's smooth because it doesn't have these striations. And we can target smooth muscle because it expresses different receptors. And smooth muscle has a certain voltage. And if this contracts, if this voltage drops, calcium floods in, contracts smooth muscle, and blood pressure goes up. So if somebody has high blood pressure, we can reduce it by blocking these channels. And we can block them with something called nifedipine. That's this chemical here. Looking at the structure here, here's a pyridine. It's a dihydropyridine because it's being reduced a bit. But these dihydropyridines form a whole class, and people decorate them with different things. They have slightly different affinities. And this is a blood pressure lowering drug. So you can lower your blood pressure by, blood pressure, as I said, increases from noradrenaline and adrenaline. So you can lower your blood pressure by reducing what we call tone. That's the sympathetic nervous system. It's an unconscious nervous system, fight or flight. And that system, if it gets stimulated, our ancestors had that working very well. They outran all these saber-toothed tigers and lived to mate, lived to leave progeny, lived to leave their genes were the products of that. So the fight or flight system was very important evolutionarily, but nowadays we can turn it on just if I sit, lie awake in bed at night and think thoughts about whether Panopto is going to work or not in my next lecture. This will raise blood pressure through this. So I can either try to do meditation and yoga and all these things, or you can pop some drugs that act on these receptors. Look at society. Most of society would rather pop drugs and do exercise and meditation and these things. Mindfulness. It's really, really useful. I'm getting more into that now. But some of these things are very effective for people that where that stuff, they're unwilling to do it or it doesn't work. So you block, the drug comes down here, dihydropyridine, nifedipine, blocks this channel. The smooth muscle doesn't have a calcium increase, it stays relaxed. Something very interesting, the relationship, flow is the easiest, but you can sort of think of flow as pressure as well. If you change the radius, it doesn't directly relate to the pressure and flow. It's radius to the power of 4. So small changes in radius can get profound effects on pressure and flow rates. So it's a very good way. It's a lot easier than trying to increase or decrease heart rate. So now we'll go into receptors. And receptors, unfortunately, diverges into 4 different ones. I'll talk about receptor-linked enzymes, ligand-gated ion channels. These are what neurotransmitters often bind to, G-protein coupled receptors, and nuclear receptors. So the first one is the insulin receptor. These in general are receptor tyrosine kinases. Rangan-Dale calls them receptor-linked enzymes. The rest of the world would probably call most of these receptor tyrosine kinases, RTKs. They're involved in growth and differentiation overall, growth factors linked to them, cytokines, the cytokine storm that's causing some people to die with COVID. It's linked to signaling molecules that go through these tyrosine kinases. And kinases mean that you will phosphorylate something. Here, the amino acid tyrosine, whose side chain is a benzene with a hydroxyl, or you can think about it like a phenol group. That phenol group gets phosphorylated. So the terminal phosphate of ATP is transferred to that. This whole group is really important. It's involved in growth and differentiation, meaning if it goes awry, you get cancer. So a lot of the anti-cancer drugs go in and try to inhibit this, either with small molecules at the active site down on the inside of the membrane, or with antibodies that bind up here and prevent them from binding. With insulin, the insulin receptor is always together. All of these receptors have 2 membrane spanning domains. The growth factor will bind up top here and bring them together. Then they can transphosphorylate one another. Insulin receptors always together, but when insulin binds, it will start enzymatic activity. And you can get a lot of phosphorylation, and hyperphosphorylation results in metabolic syndrome. We won't worry about that. Also, this one arrow here covers a lot of cell signaling, covers a lot of sins, and we're just covering that up. And I feel bad. Some people spent their whole life on probably 3 pixels of that particular arrow. We're just saying that we get transporter recruitment to the plasma membrane then. So insulin, you eat something sugary to give you context. Glucose is absorbed, goes into your blood, your pancreas secretes insulin. The insulin binds to insulin receptors, let's say on your muscle. Your skeletal muscle then will recruit vesicles containing the glucose transporter. It goes to the plasma membrane, and it will allow glucose in down its concentration gradient. That's what's going on there. The time for this is hours to minutes. What I forgot to say is, no, I didn't. I guess we're on the first one. That's fine. Look at the time here. This is hours to minutes. So that's sort of makes sense in terms of how often we eat, how often we get glucose spikes. So watch the time for all these different receptors. In terms of the next receptor class, it will be ligand-gated ion channels. These are usually activated by neurotransmitters. The neurotransmitter here is acetylcholine. This particular receptor is called the nicotinic acetylcholine receptor. It's a 5 subunit pentamer. And here the gamma subunit's removed so you can see the pore. And there's a gate and there's a selectivity filter. This one usually has good selectivity for sodium. So this will be at the end plate of muscle. So if you want to move your muscles, you're releasing acetylcholine from motor neurons. Here the target isn't another neuron, it's your actual skeletal muscle. And this nicotinic receptor will open, sodium goes in, depolarizes, and the muscle will be flooded with calcium and contract. It's called the nicotinic acetylcholine receptor because nicotine can activate it. There are other types of acetylcholine receptors called muscarinic. We are not going to touch on those. Read in Rangendale if you want to know about that. These are nicotinic ones. So somebody who is addicted to nicotine, it used to be cigarette smoking. Nowadays, it's probably vaping, how you would even get addicted to this stuff. This illustrates how you can get addicted to other things like benzodiazepines, which is a drug you might be given for anxiety or if you're not sleeping, but it's very, very addictive, just like nicotine. So with these subunits here, each subunit comes from a different gene, and then they assemble as these proteins. Acetylcholine, there's 2 binding sites indicated with these arrows. There are subunits, and there are every delta subunit isn't just a delta subunit. They start having numbers too. So there's different families of subunits. So there's subunits of subunits. If you smoke, certain neurons in your brain will think they're being overstimulated. The body always strives for homeostasis. So if it thinks it's being overstimulated through nicotinic receptors, it pulls out a delta subunit where that receptor would easily be turned on and puts in a delta subunit that makes it much harder to turn on that subunit. Now, after you've smoked and you wake up in the morning and there's no nicotine in your blood, those receptors aren't firing enough, those neurons aren't firing enough, and you feel like crap until you have more nicotine. Then it starts firing at normal rates. If you go off nicotine, it takes a while for that craving to go away because it takes a while for the cell to figure out it's got to put the old, harder to activate delta subunit in compared to the new one when you were on nicotine. This is one of the molecular theories of addiction. Trevor Sharp will tell you about a few other theories of addiction involving G-protein coupled receptors, but that's one for these ligand-gated ion channels. This receptor, as I say, it's on muscle cells as well as neurons. And there's a compound called tubocurarine. Its structure is shown here. This comes from the plant product curare. Curare is a paste made from a climbing vine in South America. And natives there learned over the ages, humans are unique in that we will sniff, taste, swallow, snort, make somebody else sniff, taste, swallow, snort something. Plant products, just were curious. And the natives there figured out through trial and error that an extract from this plant caused paralysis. So they dip their arrows and darts from their blowguns into this to hunt prey. If it nicks the prey, it shuts down all the muscles. It paralyzes them. It doesn't cross the blood-brain barrier. So these animals would be completely conscious, but it's very effective. It binds to this receptor, but rather than turning it on, it shuts it off. It blocks it. It blocks the acetylcholine from being there. In pharmacology, we call this a competitive antagonist. So tubocurarine is a competitive antagonist. Curare is this crude plant extract. Again, we throw Western medicine techniques at it, organic isolation techniques. From that, they isolated this molecule here from this crude plant extract. Tubo comes from, it was shipped in bamboo tubes. There was some that was shipped in gourds, some that was shipped in something else. And they used to think it had different properties. It was all the same stuff. This is the active ingredient. They tried that as a muscle relaxant, as an adjuvant for certain surgeries when they put people under general anesthetic. The initial trials, this might be apocryphal, it might just be urban legend, but it's kind of a cool story. Some things, even if they're not true, should be, sort of thing. That they tried it in operations and the surgeons loved it because the patient didn't move, the muscle is really springy, you can make small incisions, stretch the muscle really easily, the patient's not twitching, the patient's not complaining. But when this wore off, the patient wasn't any pleased about it because they would still feel everything. All their sensory neurons would work. They were fully conscious. They just couldn't move. They couldn't vocalize that they were in pain, that sort of thing. Don't know if it's true. We now use muscle relaxants in surgery. I'll say a little bit about that when I give you the lecture on anesthetics, local and general. What I'd like you to do is look at all these structures and think about it. Here we've got a positive charge. There must be something on this molecule that recognizes that positive charge on the receptor. This is permanently positively charged because it's a quaternary ammonium. On tubocurarine, we have a quaternary ammonium. The other one depends on pH. This is sp3 hybridized, so it's probably going to be protonated at anything under 9. 50, its pK is probably around 9, so that'll be charged. I want you guys to go away and think about this. What's the charge on nicotine? I'll cover this in detail. I'll give you guys the answer when we do drug metabolism and pharmacokinetics. But think about the lone pair electrons on these two nitrogens. The bonding is sp3 here, sp2 there, or if you want to think about molecular orbital theory, but think about how that affects pKs. How does that affect whether, when you smoke a cigarette, why do you have to inhale it? Why do pipe smokers get the nicotine effect by just holding the pipe smoke in their mouth rather than inhaling it into their lungs? So all through this course, I want you guys to look at molecules and think about what the functional groups do. What is the protonation state? Is that greasy or is that a non-greasy molecule? How's that going to affect pharmacokinetics? How's that going to affect membrane crossing? How does that affect the administration? Once we do drug metabolism, we'll throw into the mix, how does that affect metabolism as well? So the third receptor I'll talk about is a G protein coupled receptor. I'll go into a lot of details on these in the next lecture. There's about 1,000 of these in us humans. We've got about 20,000 genes, so 5% of the genome is G protein coupled receptors, but they only go through essentially 2 pathways. This cyclic GMP-C, AMP, cyclic AMP, adenosine monophosphate pathway, and the other one's a calcium pathway that's involved in contracting smooth muscle. You'll hear about that one in a subsequent lecture. So the one I'm picking on here, here's your G protein coupled receptor. It has 7 alpha helices that thread through the membrane. Biochemists would call this a 7-transmembrane receptor. We pharmacologists call it a G protein coupled receptor because it interacts with G proteins. G protein is here, it's alpha I. There's an I for inhibitory and S for stimulatory, and one called Q that activates the calcium pathway. I'll talk about that one later. Here we're dealing with the inhibitory one. It's called a G protein because it binds guanine nucleotide, not guanine nucleotides, either the diphosphate or the triphosphate. So here it is binding the diphosphate when it's inactive. When the receptor binds morphine, so morphine's from poppies, this again shows how drugs were developed and discovered. Cultures in the East took cannabis because it grew wild there. Or sorry, not cannabis. They took poppies because they grew wild there, and they would do tea extracts from it, and it would calm people down. It would help them sleep. In the West, we used to have alcohol extracts of poppy that you'd give to babies when they were cranky and they'd go into a sleep. It's called morphine, from the Greek god, son of sleep, Somnolus, I think, was sleep, and Morpheus was god of dreams and sleep and visions and that sort of thing. So that's where morphine comes from, Morpheus. So it's related to sleep, and people would drink tea. We then threw Western science at it, Western medicine, Western chemistry. We isolated morphine, and that caused a lot of problems. It caused the Opium Wars, all sorts of problems. Again, a molecule causing problems at the level of society. So you can look at, in a weird way, that this one molecule can be amplified not just by signal transduction pathways, but through how society responds to this. A molecule that caused a war almost. And once you get these isolated compounds, they're far more potent than just drinking tea from poppies. So the dose makes the drug. I'll go into more detail on that. Paracelsus, this old quote, the dose makes the drug. So morphine activates this. It goes through, it inhibits the enzyme adenylyl cyclase. This is an enzyme that takes ATP and it makes a cyclic nucleotide from it. So the 5' to 3', you get a cyclization. The phosphate's part of a 5-membered ring at that point. It's a very stable little molecule. And that molecule goes on to do many things, and its effect depends on the cell type. Here we're talking about neurons. Let's talk about pain neurons in particular. If cyclic AMP levels increase, that pain neuron is more likely to fire. If cyclic AMP levels decrease, the pain neuron is less likely to fire. In a nutshell, that's what's happening. We won't go through all the targets, but that's what's going on. So if you take morphine, it stimulates the receptor kicks off GDP, gives on GTP, that's the activated G protein. When it's activated, though, it's an inhibitor. It's putting its foot on the brake. So it's like driving around in a car with one foot on the accelerator and one on the brake. This is just pushing the brake down without changing the accelerator. We drop cyclic AMP, we drop neuron firing. If it's a pain neuron, there's far less pain. These are the most potent painkillers we have out there. There's a big jump. We go from NSAIDs, non-steroidal anti-inflammatories, to opiates, and there isn't really anything in between. I wish there was more done with cannabinoids, these types of things. I think that would be quite useful. The other thing, well, I'll tell you more about this tomorrow when I go through some of it. Yeah, I might leave it at that. This time scale is seconds to minutes. Some of the effects of adrenaline are on adrenaline receptors. And these are G protein coupled receptors. So if you get frightened, this fight or flight response, that will increase within seconds and stay up for 30 seconds or minutes. That's the type of time scale we're looking at with these G protein coupled receptors. So now we move on to nuclear receptors. So with nuclear receptors, as the name suggests, these make it all the way to the nucleus. These are all based on a steroid nucleus. So you've got cholesterol, that forms a steroid nucleus, which is these 3 cyclohexyl rings and 1 cyclopentyl ring. That's a steroid nucleus. And then it gets decorated. It gets very Baroque. You pull things off, you put things on. Nature did that. We medicinal chemists do a lot of that. From these operate by, because they're greasy, they're made on cholesterol, which is a component of the membrane. They will cross through membranes very easily. They'll get into the cell, they'll get into the nuclear, through the nuclear membrane. They then bind to their protein receptor, and this is a transcription factor. It binds to DNA, and either opens it up and allows RNA polymerase to make RNA from that, which then gets exported and made into protein, or they bind to their protein. The protein binds to the DNA and it prevents separation of the DNA strands prevents RNA polymerase from binding to that. So they can be positive or negative. They can be activators or repressors. 3 examples we'll go through. 2 are sex hormones. So you can see that these things are reasonably similar, but they have profoundly different effects in terms of the physiology. Going through puberty, these take years. So here, time, hours, days, months, years, because they're making protein changes. They can do profound things. Puberty, the onset of puberty till fully developed is probably several years. Also these days, if people are unhappy about their birth sex and they want to become a different gender, these are the types of molecules that allow that process to take place. So they're very, very powerful molecules in terms of affecting the physiology of a person. I'm going to talk about progesterone a bit. So progesterone is one of the 2 female sex hormones that's involved with a woman's monthly cycle. Progesterone, though, they thought about progesterone will stop ovaries from developing. So you could give this as a birth control, but you'd have to inject it. Nobody really wants to inject themselves every day. You have to do that with insulin, but that's a different matter. So progesterone wasn't effective orally because it gets metabolized up at, well, I might as well show it, up at this position. It's called the 17 position. It gets oxidized. It can be converted even into something that looks like this. This is a male hormone. It's related to testosterone. Progesterone was modified by putting a big bulky steric group on here on the 17 position. So the enzyme's mouth, its teeth couldn't get on it to oxidize it. And it was one of the first orally bioavailable birth control methods. There is also estrogen. So estrogen is produced by the developing egg. I'm forgetting all this. I think progesterone is by the follicle. And that will also stop development, because if you're already pregnant, you don't want to have another egg started at that point. So estrogen levels spike and they'll prevent other eggs from developing. The other birth control pill was developed on the basis of estrogen. Same problem, it got metabolized, so they put something here. That whole story, again, molecules impact on culture in tremendous ways. I don't have time to go into all of it, but read some background material on the development of the pill. It's fascinating at a molecular level, physiology level, chemistry level, molecular level, physiology, and societal level. At this point, I'd usually look around the audience, and it's usually about 65% women in this class, which is starting to reflect the overall distribution of Oxford students. And all the women here would not probably be here, some people argue, if it wasn't for the pill. The pill gave women control over their sexuality and when they got pregnant and when they chose to get pregnant. And from that came women's rights, sexual revolution in the 60s. All of this can be traced back arguably to the pill. There are whole books on it. Carl Djerassi, Djerasi, something like that. He was involved in the development. At the molecular level, this structure, you could start with cholesterol, but it would be a lot of steps. There were a couple guys who worked in the chemistry department in Oxford back in the day. I think it was Birch reduction, and Cornforth, who did a lot of work on steroids and synthetic mechanisms and converting them. It wasn't total synthesis. You'd usually isolate things from plants and then monkey around with them, toy with them at that point. But nobody could make progesterone at the high levels that they wanted to for trying to research this, or even indeed rolling it out as a birth control pill. And then there was one guy in the States, I think his name was Market, his last name was Market, and he developed what's called the Market process, and he patented it, aspects of it, not the whole thing, aspects of it. And he found that there was a plant sterol in a species called Dioscorea. Dioscorea Mexicana has a lot of this, and as the name suggests, this is a plant in Mexico. Dioscorea is a yam. So yams are all over the world. In the States and Canada, we call sweet potatoes yams, but they're not Dioscorea. It's a different genus. But anyway, this one in Mexico had a high level of a compound called dioscogenin. I don't know how to pronounce that. Dioscogenin, something like that. And this market guy had a 3-step synthesis to progesterone. And he isolated it and he made kilos of it. And then he went to a drug company, a pharmaceutical company in Mexico City called Syntex. And they dominated the world supply of all steroids and steroid precursors. And then he got into a spat with people there and he left and he didn't give them the full secret of how to do the conversions. And this is where this Carl Djerassi was hired. And he went there, developed the pill, really was a strong proponent of it. Djerassi is like a Renaissance man. He wrote a lot of plays about science, about the role of chemistry in science. Fascinating stuff. He's sadly dead now, I think 4 autobiographies. Not a small ego, but a very, very smart man. So read around the story of progesterone from the chemical pharmacology, the med chem stuff, and the societal stuff. Very interesting. In terms of anabolic steroids, if we just took testosterone, so this is an analog of testosterone, it would have 2 types of effects. One are androgen effects, meaning eliciting secondary male sex characteristics, those being facial hair, male pattern baldness, unfortunately, muscularization, Adam's apple, deepening of the voice, these types of secondary male characteristics. So those are androgen. The other side of it is anabolic. Anabolic meaning building up muscles. This is where people start taking anabolic steroids. They weren't originally designed for drug abuse in sporting events. Anabolic steroids were designed for people that were bedridden and you don't want them to lose muscle mass. You or one of your friends may have injured your arm and you get in a cast. You take the cast off, your muscle is atrophied a lot. It comes back quite quickly. Some people are bedridden for a long time and you want to keep muscle mass on them. That's why they developed anabolic steroids. They do the anabolic side of things without the androgen side of things. Back in the day when bodybuilders and strength athletes who were doping just took testosterone, they would get male secondary sex characteristics, including females who were doing this. If you read about some of the atrocities in the former East Germany, they were giving all their female athletes what they thought were what they called vitamins, and they were laced with old generation anabolic steroids that had a lot of these inducing male secondary sex characteristics. And there was a class action lawsuit with that as well. So this is sports doping. And there was always a competition between the new anabolic steroid that couldn't be tested for and ones that you could test for. So there's an arms race with this. At this point, when I was going through the lecture, I was very tempted to put something in here rather than nandrolone, which is an anabolic steroid, stanozolol. And I like that one, kind of. I like the stories around it, I guess. It relates to a guy called Ben Johnson. All of you guys are probably way too young to know this. There was a 1988 Summer Olympics in Seoul, South Korea. And Ben Johnson was a sprinter, world-class sprinter from Canada. And he won the gold in that, beat everybody by a long way. He was even looking over his shoulder when he passed the finish line to see where the hell everybody was. Day later, he got caught drug doping, and they found stanozolol in his system, in his urine. The problem was he was doping. There was a big inquiry in Canada over that. And he was definitely doping, but everybody was doping. Other than Carl Lewis, the American in that race, claims he wasn't. We'll never know. Everybody else in that race was eventually found or admitted to doping with steroids as well. So it's kind of sad that Ben lost his medal, but that's life. What's interesting, though, is Ben was doping with a different steroid. He was not doping with stanozolol. He was doping with something that has, down in this bit of the molecule, instead of, I can't remember what stanozolol has down there. He had one with a furazo ring, so a furan ring with nitrogens in it. That was his drug of choice. So he couldn't figure out how he had this other stuff in his system. Probably somebody slipped it in his water bottle, or he had to drink beer after the race to generate enough urine to do the urine test. So again, from compounds all the way up. Also, don't, it's one little aside, another side. Our generation in memory sort of isn't the first one to dope with drugs for athletic performance. The ancient Greeks took 5-HT2A agonists. They didn't know they were. They took magic mushrooms. It's like taking LSD or psilocybin before they competed. The ancient Romans, gladiators, ate extracts, crushed up testicles from cows, from bulls, and they would have got a lot of testosterone for that. So doping goes back a long way. This last one here, it falls into a class called glucocorticoids. The chemical I'm showing you is beclomethasone or beclometazone. There's a prodrug and a drug. It's not clear to me which is which. The chemical structure is definitive, though. Look at these isopropyl groups, or sorry, N-propyl groups out here put on the molecule. That makes it more greasy. So this probably makes it orally bioavailable. In the anabolic steroids that Ben Johnson and these other people are taking, they're also modified at this 17 position, so they don't get modified. If they don't get modified, when you swallow them, you can take them orally. All the other anabolic steroids, like this nandrolone, has to be injected. You usually get an injection in your thigh or your butt, your gluteus. These glucocorticoids are anti-inflammatories. That explains this weird name I told you earlier, Nsaids, non-steroidal anti-inflammatories, Nsaids. Why would you name a class of drugs for something that they aren't? It's because the glucocorticoids are anti-inflammatory. So everybody knows that. They downregulate all the enzymes. They downregulate cyclooxygenase. They downregulate the enzyme that clips off arachidonic acid called phospholipase. Lipase cleaves it, hydrolyzes it, A2, so that's the A2 position where arachidonic acid is. It just downregulates all those at the level of genes. They're very powerful. The problem with these drugs are that they have powerful effects. The theme here is that these all have profound effects on the appearance, the phenotype of people. If you go on glucocorticoids for a long period of time, you get what's called Cushing's syndrome. You can get it from certain cancers in certain glands, or you can just be on glucocorticoids too high a dose for too long. And it's a bit unkind, but it's easy to remember. You get moon face, and if you take a marshmallow and put toothpicks in it, you look like a toothpick marshmallow man, because these glucocorticoids take all the muscle, they metabolize all the muscle and fat from your appendages and put it onto your trunk, onto your body. So you look like a marshmallow man. So you can't keep people on this very long. So for rheumatoid arthritis, it's really effective, but the side effects are too great. The other side effect is it weakens the bones. You get a lot of osteoporosis with this particular one. This is just an illustration showing you that drug targets occur in families. This is adrenergic receptors. I've already told you about some of these adrenergic receptors. They're expressed different ones, the different isoforms in different tissues. So the smooth muscle around the vasculature has alpha-1. So if we do an alpha-1 antagonist, that's another way that we can have a blood pressure lowering drug. So prazosin is a blood pressure lowering drug. We can have beta-1 on the heart, and we can slow the heart down from an adrenaline rush. We can make the heart beat more slowly with atenolol. This is an antagonist of that. For the lung, there's a beta-2 adrenergic receptor. Weirdly, well, I guess you guys wouldn't know that it's weird. Beta-2 will relax smooth muscle in the lung. So something like salbutamol relaxes smooth muscle. Salbutamol, if you have asthma or your friends have asthma, this is what it's doing. It's going through one of these signal transduction pathways, and from the beta-adrenergic receptor down through that cyclic AMP and protein kinase A. And if, let's say we were in the liver, all these pathways are the same in different cells up to the last couple steps. If we were in the liver and we said we were releasing glucose from glycogen, glycogen's a polymer of glucose, glycogen phosphorylase does that. So from adrenaline through its receptor, cyclic AMP, the second messenger, all the way down, there'd be 6 Nobel Prizes. This is how important these signaling pathways were. That signaling pathway occurs in the lung, occurs in smooth muscle, and it relaxes my smooth muscle. I have asthma, so every day I inhale something that activates this receptor and allows me to breathe. The overall idea of this is if we've got different receptors, we can tweak a molecule chemically, we can do medicinal chemistry on it and get receptors and target different receptors selectively. Here I have atenolol. That's a derivative of something called propranolol. A guy called Black, James Black, got a Nobel Prize for working out beta antagonists from adrenaline. That's how that worked out. So this whole idea, it doesn't matter what your target is, there will probably be variations in that target. They'll probably be expressed differently in different tissues, and you can probably make small molecules that selectively target them. That's drug discovery in a nutshell. What I'd like to do now is just talk about some of the control systems. So wherever there's chemicals, we can intervene with small molecules and perform pharmacology. So we have a central nervous system and a peripheral nervous system. I showed you from that blood-brain barrier stuff with the trypan blue. The central nervous system separate from the peripheral. All the nerves, though, use this gap. They use neurotransmitters. So we can make things that act like neurotransmitters, block neurotransmitters, make more neurotransmitter go out, block reuptake of the neurotransmitter so the steady state increases, gives us a lot of levers to pull with this. So that's been the sites of many, many drugs that were designed specifically for the targets or found accidentally through society just eating products. Another system that I've talked about already, adrenaline being this hormone, it's a traditional hormone, adrenaline. So adrenaline is, traditional hormone means it's produced in one cell, secreted into the blood supply, and then leaks out and acts at a distant cell. Nowadays, the world's a little more complicated. We have autocrine messengers. If you have one cell, it can spit something out and stimulate itself. It can stimulate the cell next to it without going through the blood, paracrine signaling. All of these, though, the hormones, they're small molecules. We can look at them. We can tweak them. We can either upregulate or downregulate the process. Even things like the steroid hormones, we can actually give the hormone itself. We might have to modify it so it's biologically bioavailable through an oral route. This is what hormones look like. I'll say more about peptide hormones later, but insulin's 50 amino acids. Cholesterol, I've talked a lot about that with the steroid nucleus. And then other amino acids like tyrosine, we can get hormones like thyroxine. This comes from the thyroid gland. This is why if you're not getting enough iodine, you get what's called goiter. Your thyroid gland will greatly increase. And it increases because it's trying to make this compound here, thyroxine. It's one of the, I think it's the only compound I know endogenously that the body actually makes that has a halogen in it. So you'll see a lot of halogens in other compounds. Usually that's a trigger, a flag that it's being made synthetically rather than a natural derivative if it has any of the halogens in it. So fluoro, chloro, sometimes bromo, sometimes iodo, but here's an endogenous one with iodine. So a lot of amino acids go to neurotransmitters, some of them go to hormones. Last thing I'd like you to think about is here. So I've been talking about targets and this traditional idea of targets. Now we know that there's protein-protein interactions. And in cells, a lot of the signaling isn't just second messengers. It's one protein binding to another protein that then recruits another protein and recruits it to the plasma membrane. Then it signals to the nucleus and transcription factors. It's all protein-protein interactions. But these are greasy bits that interact. How can, it used to be, they'd say we can't target them. Now we think, okay, we can target a few of them, but not in general. I'm hopeful that you guys might make me proud. Go out there and figure out ways of a general method of doing this. This also has implications for diseases. So COVID is an RNA virus. You have DNA viruses, you have RNA viruses, you have bacteria that are living cells. They don't even need our host apparatus. All of these things are infectious because of the DNA, especially viruses. It's DNA or RNA. There's an infectious agent that only requires a protein, and they're called prions. That's an abbreviation of proteinaceous infectious particle. The cellular form has, this is what it looks like actually. It's got 4 alpha helices that are overlaid, and it converts to 2 of those alpha helices go to 4 beta sheets. It exposes hydrophobic amino acids on the side. This can then form an aggregate. This aggregate builds up and it gives you diseases such as mad cow disease, bovine spongiform encephalopathy. It will also give you diseases such as scrapie in sheep. It's sheep to sheep transfer. That hasn't come into man yet. Unfortunately, mad cow has been into man. It only, thankfully, only affected, I think, between 170 and 200 people died from it. So it was contained. This gives us an idea that aggregating proteins are very detrimental. And if we have ways, if we can figure out ways of getting rid of the aggregate or preventing it, we might be able to prevent disease. This was a very controversial idea that prions could cause disease. Stanley Prusner, a professor at Stanford, got a Nobel Prize for this several years ago. Some people still don't accept that it's prions. They say that actually there's some DNA or RNA that we're just not seeing. The more broad prospect of this is in, at the bottom here, Alzheimer's disease. So in Alzheimer's disease, you get beta amyloid plaques. They follow the same thing, greasy bits sticking together, can't degrade proteins. In Parkinson's, we get something called alpha-synuclein that also aggregates. If you got a way of getting rid of these, you might be able to treat the disease, Alzheimer's in particular. This is where I will end on this lecture. I can never pronounce these antibodies. They all end in umab, aducanumab. That's my attempt at it. This is an antibody, so antibodies are Y-shaped, against the soluble form of beta amyloid. They gave the antibody intravenously. How the hell it crossed the blood-brain barrier, I don't know. It dropped beta amyloid plaque levels. We can measure that with an imaging technique called positron emission tomography. It dropped them a lot. Fortunately, it didn't improve cognitive function according to some people. The pharmaceutical company Biogen says, yes, it did improve cognitive function. I don't know. We'll have to see. I really don't know on this. It's very topical. This was out just in November. The FDA didn't approve it, but Biogen's going back to try to get a more narrow classification. We'll see. Anyway, see you at the next lecture.